Welcome everyone to the Symposium Knowledge Graphs for Plant and Microbiome Multiomics. First, I would like to thank the Fair Data Fund of 40U Research Data for sponsoring this symposium today. And of course, thanks to my supervisors for the support and to the speakers. So in this first presentation over a few minutes, I will tell you just a little bit about the work that I do and introduce loosely the topics of the symposium. But then I hope that the speakers will deep dive in the specific topics and tools that they are actually experts of. So my name is Elena Del Poop. I just started my third year PhD here in Wageningen at the Bioinformatics group, together with Marnix Medema and Justin van der Hoofd. So in my PhD, I'm working in integrating omics data to discover the hidden biochemical potential that is there in plants. So in this short introductory presentation, I will largely speak about plants, but I'm going to introduce a bit of the work that I do and also the overall, why I think knowledge graphs can be one of the key tools to solve some of the challenges that are there in this field. And also I will present to you the vision that I would like to realize by the end of my PhD, which is to create a linked knowledge base for natural product discovery in plants to enable community-driven curation of the plant biochemical space. So, but why am I working with plants? So plants are the best chemists on the planet. And that is because they are sessile organisms, meaning that actually to cope with the stressors that they encounter, they don't have any other resource than their biochemical artillery and biochemical resources. And that is why actually plants contain the largest library of natural products and bioactive molecules that we can find among other kingdoms. And because of these very interesting ecological functions that these compounds can have, we find applications ranging from medicine to agriculture to sustainable materials. But so, however, to truly make sense and to truly make use of these interesting bioactive compounds, we need to know how they're made, meaning their biosynthesis. And the biosynthesis of these compounds is, of course, encoded in the plant genomes and in the expression of these genomes. But because of the complexity of plant genomic organization, we actually have an extremely fragmented and largely missing knowledge about this biosynthesis. And we miss the links then with the final chemistry of the final compound. So how do we overcome this bottleneck of connecting the chemistry to the biosynthesis and just getting to know more about the biosynthesis of these specialized compounds? So nowadays, we have many different resources across different omics, from genomics to transcriptomics, but also the regulatory environment, meaning the different regulators that actually tell us something about gene expression, as well as the phylogenetic relationships and evolutionary conservation. But given such a very heterogeneous landscape of different data that we can combine together and explore together, the biggest challenge that we're facing right now is how to link these data together to make sense of it. So how do we link these data? So as we see that more and more data sets are becoming available across different omics, how to link it and explore it becomes the biggest challenge. And this is why one of the solutions that is available is actually revolves around the topic of this symposium, and that is knowledge graphs. So knowledge graphs are a simple way to organize knowledge as a network of relationships, where every piece of data is a node, and the relationships between these different pieces of data are represented with ontologies and semantics that help us really define these relationships in a systematic and easily explorable way. So, so this means that a computer can not just understand data, but also understand how data relates to each other. And thanks to knowledge graph, actually, its use in the natural product space is that we can actually combine different resources together and make the data easily explorable link it with public knowledge, make it retrievable, and actually like aid in exploring it and link it with other evidence, ranging from phylogenetics, the biological like taxon, for instance, or the chemical structure. And some of the speakers today are actually working on some of the key tools to make this possible, from Wikidata to Wikipathways. And I hope that they are going to tell you more about these things just in a little bit. Actually then like transforming the data in like knowledge graphs, or making it available as a knowledge graph, helps us to increase the value of the data, because each data set doesn't live anymore in isolation, but its value is increased by being connected with a larger web of knowledge. So, and this is why the vision that I would like to realize by the end of my PhD, and with specific application to plants as my topic, is to make it possible to increase the value of the data that is generated in multiomics, and link it, for instance, with metabolomics datasets in plants, by integrating new datasets coming in from different users together with publicly available knowledge. And then use knowledge graphs to also standardize the way that we annotate this data and we enrich it by connecting it with the other available databases. And by doing that, to be able to create a community-driven way to annotate the plant biochemical space, both by recording what is the experimentally validated knowledge and link it with other databases, but as well help creating a community-driven way to generate predictions, and also to rank them and prioritize them based on evidence. And when I mean predictions in this case, and in what I'm doing in my PhD, I mean predictions of biosynthetic pathways in plants. So this is, of course, a very ambitious vision. I'm happy to be taking together with you the first step in this symposium by being here together and trying to brainstorm around how to make this possible and how to connect this with other resources that are available in the community. I want to mention that thanks to the Fair Data Fund, we didn't only support the organization of today, but I actually had the chance over the summer to work together with 2 other student assistants on 2 different small summer projects that will be key steps for achieving this vision. One of them is Max, and he helped me to actually translate the experimentally validated knowledge in plant biosynthesis from the database Plant Metabolic Network. You might have heard of it also as PlantPsych, and translate it into graphical pathway markup language. That is the format that is needed for Wikipathways. You are going to hear more about Wikipathways in the last 2 presentations today. And the goal of that is that it makes plant pathway information easily queryable, and it makes it very easy to be linked with other chemical databases, as well as also integrate links with other databases and resources in the pathway space, such as MyBig, as well as Plantismash, which are 2 resources for genomically clustered pathways. Another student project that I carried out during the summer was together with Ariel. And in this case, we translated the public metabolomics data that is available from the PanRedo database we made it ready with ontologies and semantics so that it can be translated in a knowledge graph format. And hopefully in the future that will make it possible also to link that publicly available knowledge with other omics datasets and rely also on metadata, for instance, to create links between metabolomics, transcriptomics, as well as other omics. So, of course, all of this is very ambitious. I'm really happy to be here with you today. One of the biggest challenges of knowledge graphs is that they really require an agreement on ontologies and semantics, and coordination also on the interoperability of the different tools that are supposed to be linked in the system. And this is why it's extremely key to gather, or I thought it was extremely important to gather a community so that we could actually agree on the standards and best practices to make this possible. And as well, like I hope together with you here in the audience to have like different communities represented. That would be great to have together, like including like future users of whatever is gonna come up from these projects, people that are generating data across different omics, as well as tool developers. And some of them are the speakers that are gonna be here today. So thank you for being here, and I'm happy to take any questions.
Questions for Elena?
You said that you wanted to do some predictions, and yeah, I worked with graph neural networks in the past, so I was wondering whether you wanted to go down that avenue or...
I would love to in the next 2 years. I'm right now, I just started some brainstormings on the topic, but I'm also looking into reading more on that, yeah.
Did you also consider knowledge graph embeddings?
Like using embeddings within the knowledge graph, or embedding parts of the knowledge graph?
I'm not sure whether that's the right name to call them, but there are also like specific algorithms for link prediction for knowledge graphs as well. And you can play around with the different relations there as well.
Yeah, that'd be great. Let's chat more later. Denise?
So in these public repositories, did you see how good the metabolites in Plantram are annotated, right? Because I think the diversity is so much larger than in humans, for example.
Sorry again, so how it is annotated, the diversity, you mean of species?
No, so of the metabolites.
Uh-huh.
I think compared to humans, plants have so many 10,000, 100,000 more metabolites, and annotations are always a big bottleneck. So how good are the annotations of individual compounds in these online databases for plant metabolites?
That is something that I didn't assess in depth, given I've been more focusing on the biosynthesis. But maybe some of these speakers have a better answer to this. I think lots of them are working with this, but maybe we're going to hear more in their presentations as well. Yeah. Well, without further ado, I think we can move to the next speaker. You have this. Yes, great. Yes, thank you. I think it was turned off already.
Yeah. Thank you, Elena. And thanks a lot for all the hard work that went into organizing this wonderful symposium. And I already look forward to all the interesting insights and discussions for today. So it's a privilege to present also briefly on behalf of Felicia Wolters, who's there also in the audience, who also is doing a PhD project here in Wageningen on multi-omics based pathway discovery. And yeah, so Felicia is really intrigued by how the evolution, how evolution has led to diversification of indeed this enormous diversity of plant metabolites. I think Denise already mentioned it. Compared to many organisms, the numbers and diversity of plant chemicals are really enormous. And this is also because of their rapid evolution and their combinatorial evolution, where you see a number of common themes in their chemistry. But actually, these are constantly combinatorialized by modifications which are enacted by modifying enzymes that are modifying these molecules into lots of different, basically varieties that each have different biological activities and ecological functions. And one of the key challenges for this field is how can we actually find out, like, what is the genetic basis for this incredible evolutionary diversification of plant metabolism. And for that, indeed, connecting these different levels, the genome to the metabolome, also via the transcriptome, and also taking into account genetics and evolution, including genome structure and the evolution of genome structure, provides really amazing possibilities. And Elena already alluded to the role that knowledge graphs can play in this, where indeed these knowledge graphs allow you to connect all of these layers explicitly and identifying the specific connections where there is actually knowledge about those connections, which can then also be used, for example, for pathway predictions of connecting all these specific varieties of these chemical molecules to specific genes and genetic variants that you see in the genome. And when you do this, metadata and context is really crucial. And this is especially the case because if you're doing this cross-species, if you're looking at the evolution of biochemical diversity, you have to compare different species, and of course their morphologies, they look different, their development is different, and you're going to need all that context to be able to figure out how do you compare one species to another at a given time point in an experiment. You're going to know, okay, you're going to need to know and you're going to need to be able to describe accurately, like what kind of tissue are we talking about, what kind of developmental stage are we talking about, because only then are you really able to make those connections also across species. So there's quite some ontological frameworks already specifically developed also for plants, and this is also something that Felicia has been using to describe the experimental data that she's been generating, and that I will tell you a bit more about in a moment. And many of these plant-focused ontologies also are really useful to describe, indeed, such contextual metadata that is crucial for cross-species analyses and comparisons. So to make this concrete, let's say you have two species and you're growing them in the lab, and yeah, maybe one actually grows much quicker than the other. Then still you want to be able to compare them, for example, between species A and species B across conditions. Well, maybe they also respond differently to the conditions. Their tissues look differently, so the sampling there is challenging. And yeah, to be able to capture this well, you need to capture all of this also inside the knowledge graphs by the right metadata. And I already mentioned that Felicia has been working on a very large data set, and this is actually where this has been put into practice for a number of species from the Brassicaceae family, which is a very well-known plant family that actually includes many famous crops like broccoli and Brussels sprout and cauliflower, etc. So this is a very diverse, evolutionary diverse family, including both wild and domesticated species, with a lot of metabolic diversity, where already quite a bit of this metabolic diversity has been studied also before. So this also provides us with quite some context if you want to use this, for example, for pathway discovery, well, whether you're actually finding or predicting connections that are real, because there is some knowledge to begin with. So for these species, Felicia generated now two very large data sets of paired omics data, where there is both transcriptome and metabolome, also connected, of course, to the genome. And the first data set is across 17 species, and this is a profiling data set, so it's really looking at mapping the entire diversity across multiple tissues, roots, leaves, and in a number of cases also flowers when feasible. And then in a second larger experiment, she zoomed into a subset of 10 species where she got condition-specific time series data with multiple conditions, multiple time points, to get even more variation, to be able to look at the covariation of metabolites and transcripts. So this is data set 1. Where 17 species were taken across the Brassicaceae, also covering the major clades in the Brassicaceae family and some outgroups as well. And this also covers some major evolutionary events like whole genome duplications to be able to also see how these pathways have evolved across the family. So this is the second data set, and these are the 10 species that were zoomed into, also because these are also working well in the lab under the conditions. So the idea was also for the second experiment that the experiment was carried out on a very standardized condition. So you want to keep as much as possible the context the same. So grow them all in the same way and under the same conditions, in the same climate, etc. So that the variation that you then introduce through the conditions, time points, different tissue sampling, that this is really strictly connected to those basically dimensions of variation that you're introducing into the experiment. And then the idea is that indeed we have multiple different species, multiple conditions, multiple time points, multiple tissues, and also replicates. And across each of those dimensions, there's going to be variation. So there's going to be variation of expression of genes and also metabolites that are produced accordingly. And then that allows you to also make connections between transcripts and metabolites by seeing, okay, whenever transcripts are expressed in specific time points, specific conditions, tissues, and species, then you're bound to also see metabolites being produced as a result of that gene expression. And this then allows, using, for example, the knowledge graphs, to make the connections between all of these different chemical variants of these molecules and the genes. Okay, so Felicia has done a lot of work also to indeed take this data set, these two large data sets, and also describe everything according to the FAIR data standards, including all the metadata that are crucial for creating these knowledge graphs. And this is also something that we hope to also get out of the further interactions, of course, with the speakers, is how we can then further also connect it to, for example, the experimental natural products knowledge graph that has been generated by some of our guest speakers. So looking forward to further discussions there. And thanks specifically to Felicia and of course to the whole team that was involved, including the people from biosystematics, like Klaas Baumeister and Eric Schanz, and the metabolomics facilities. Okay, thank you.
How to know if the recording is on? Do you know?
It's always on.
It's always on? Wow. So we invite Louis Felix. I'm gonna put your slides on.
Yes.
Just checking.
And if anybody would like to discuss it or hear more about the data set, you can also talk to Felicia in the break.
Is this that there is like the assessment? There's something
Yes.
That's right. Because I'm interested. Yes, I think so. Yes. Yes.
Thank you. All right. Thank you very much. Thank you for the invitation. And it's a pleasure to be with you today, presenting some of our latest work regarding the use of large language models and knowledge graphs. This presentation was designed in two parts. The first is on a prototype for a multi-agent system using LLM to facilitate the exploration of mass spectrometry data in metabolomics. The second is ongoing work to leverage large language model and scientific literature for accelerating exploration of scientific articles and prior knowledge. All right, let's start. So I wasn't sure about the composition of the audience, so I wanted to start from the very beginning. What is metabolomics, and specifically mass spectrometry-based metabolomics? If we summarize it in few steps, we will start from a biological sample or environmental sample. Here it would be a plant. The metabolites would be extracted, typically with organic solvents to have this extract. And this extract is analyzed on a mass spectrometer, often coupled with liquid chromatography. And essentially what we are doing, we are separating the molecules based on their properties. And we would be, is it the later? Can I use this? Not too fast.
Or the pointer should be on the...
Is it?
Also on the controller.
Laser pointer. Yeah, but it's delayed. Yeah. Thank you. Essentially, we'll be generating fingerprints for the metabolites that are detected as ions. And these fingerprints are mass spectra, fragmentation spectra, where the molecule is fragmented into pieces. And we can use these pieces, these fragments, to propose the annotation of structural properties. And mass spectrometry is limited because we cannot determine full chemical structure. We can only establish some of its, let's say, 2D properties. And because of that, we rely on many different computational tools in particular. So you might know some of them. But essentially, we will need to rely on a battery of different softwares to annotate these spectra and to propose molecular formulas, chemical structures, chemical classes, and this is the process of the annotation. And this process is quite complex. So if we want to propose hypotheses, there is a need to look at the different dimension of the spectra that were collected. So we need to look at the signal, we need to look at the annotation, at what species we are working on, and etc. And we have this concept in natural products that was proposed by Bill Gerwick of having the different facets of a molecule in order to understand its structure and its function. And really, not only experimental data is needed, but also we need to access all the prior knowledge. And for that, we have the chance, thanks to the organizers, to have participants of the speakers of this seminar that contributed to the design of databases that are mapping the link between metabolites and species. So we can look at, okay, I work with a brassica plant. I can collect what was discovered in the past, and I will use this to try to make sense of the experimental data and computational annotation that we produced. Yes. And one of the emerging ways to represent a complex heterogeneous set of datasets or information is the use of knowledge graph and the purpose of this meeting. So I will not describe in too much details on what is knowledge graph, but essentially it's a semantic representation of complex information. And Pierre-Marie Allard is the one who introduced this concept for natural product chemistry. And in this paper, still recent paper, he was able to propose a framework to represent the experimental data collected on mass spectrometry-based metabolomics into a comprehensive and detailed knowledge graph encapsulating both information about the biological species all the way to the spectra and their annotation collected, and down to the bioactivity actually of some of the compounds. So this is the foundational framework that we can leverage to integrate this complex information. However, navigating these knowledge graphs is very challenging. We can use the Sparkle language to query information from different interactions. And in order to achieve that, one needs to be an expert in the Sparkle language. And so you will see on the left side that what we observed is that the people that are expert in Sparkle, they are not expert in natural product chemistry. There are some in the room. Typically, there is a gap in skills that is preventing the leveraging of such capability for application in research. And this was very pronounced when I started my postdoc position with the professor Jean-Luc Wolfender in Geneva. And during this project that was initiated by Pierre-Marie Allard as well, the project aim was to analyze the largest plant collection in the world. And this collection was analyzed by LCMS, mass spectrometry-based metabolomics, and a knowledge graph was produced using the framework that I've just mentioned. And when this collection of data was achieved, we encountered this issue that the people who had the ideas about, can you look into this hypothesis? Can you look about these computational aspects of the data, the signals and so on? They were not able to produce Sparkle query, and so this was created a discrepancy, and it was slowing down the entire process. And this happened in 2023, yes, at the end of 2022. And then ChatGPT was just released essentially. And so we thought, okay, can we start to use large language models to accelerate? No, it was, yeah. And so they changed the model rapidly. At the start, it wasn't functioning. But then after the months with the release of 3.5, we started to see some ability to produce meaningful queries. And with some engineering, so you might have heard of, so I presume everyone is familiar with large language models, okay? And also with the concept of prompt engineering, context enrichment, and so on. I will get back to this in more details. Essentially, if we provide the required information when asking the model to produce an output, it will perform much better if the required information are described in the query. And so, empirically, there was an intuition, an observation that it was possible to achieve something. And even down, all the way down to the entire process to interpret the results of the Sparkle output that was returning from the knowledge graph. And when I started my research group at the CNRS in France, so in 2023, we had this idea of combining the expertise of the web, semantic web, with the Dr. Fabien Gordon at the INRIA, it's the Informatic Institute in France, and the expertise on natural products with the Professor Jean-Luc Wolfender to create a system that could automate the production of Sparkle queries for mining this experimental knowledge graph. Okay? And so with some of the lab members, we designed this multi-agent system. So a multi-agent system, it's an informatic system that is composed of different agents. Here, the agents are large language models. Okay? And so in this overview, you can imagine having the researcher, the user, asking a question in natural language. Okay? And we will have a first large language model agent that will be tasked of checking is that question relevant? Is that relevant to the knowledge graph? Because some people were like stress testing the system, asking a political question and so on. So there is a need to first check the question, verify that it was formulated with sufficient detail. And so if needed, this agent can ask clarification to the user. Okay? If the question was valid, it is given to a supervisor agent that will then distribute tasks or actions to specialized agents. One that knows how to interact with the knowledge graph, another one that is specialized in generating a Sparkle query, and so on. And the system was designed in such a way that it can access a Sparkle endpoint, test the query, get the output, and see and provide some first interpretation back to the user. So more concretely, we would have the input question here, the entry agent, the validator that I've just mentioned. And here, the supervisor will first need to collect the entities IDs. So as soon as we start to discuss knowledge graphs, we have to think about ontologies, we have to think about entities IDs and so on. And so to translate the natural language questions into a Sparkle query, we need to collect all these entities IDs. We need to access the schema, so the description of the knowledge graph, and to assemble all these building blocks in order to produce the right Sparkle query. Okay? So entities are collected, they are resolved, the Sparkle query is applied, and so on. And so each agent, and so you might have heard of this as well, has a set of tools available to it. So tools would be, for example, a Python interpreter, or it would be a retriever for some information stored in a database, and so on. So this system is redesigned around LLM agents and a set of tools that they can be leveraged depending on the input question to collect the required information and to achieve the end goal of providing to the user the results of the Sparkle query and some basic interpretation. Okay? How do we evaluate such a system? It's very complex. So we had several attempts in achieving that. As always, we need to have a benchmark. A benchmark here would be a set of natural language questions and target Sparkle queries. We crafted them in a way that they were categorized from easy to very complex questions. And then we tested the system in different configurations. So either one-shot LLM configurations, like ChatGPT with with all the required information, so entities IDs, schema, description of the knowledge graph, and so on. So it's one-shot test. Then all the agents configured with a medium performance LLM model, and one with a high performance one. And so the results that I'm presenting here, all that is part of the preprint. Essentially, you will see the three configurations here and the different categories of questions. And what was observed is that one-shot LLM is not capable of solving complex questions. It might, with some prompt engineering, it might work. Okay? This is not an automated process anyway. The multi-agent system starts to be slightly better with the medium performance model, but it's only high performance model that were able to achieve a relatively high level of resolution for the Sparkle query. And just for the anecdote, 3.5 was not at that level. So GPT-4 was really a significant gain in performance. So such a system required to be crafted beyond just a single LLM. It required to have the tools, and it required also high performance LLM. So such approach wasn't feasible 2 years ago. This project was, let's say, at the start of a larger initiative, the Metabolic AI project. And this was the prototype, the proof of concept for this French and Swiss project that started recently. The archetype objective is to design an AI scientist for interacting with knowledge graphs for metabolomics. Okay? And so we have a lot in common with the work you are doing here and the project you are working on. So I see a lot of synergies and complementarity, and that's good that we are coordinating at the start of our project. But essentially, it would encapsulate mass spectrometry down to the scientific literature. And through a unified knowledge graph framework, so we are talking about alignment, the goal is to interconnect this with an LLM system that could accelerate data mining and possibly hypothesis generation down to some advanced human-computer interface capabilities to visualize the results and to provide some interactive framework. Okay. So this project has a large number of partners from both French and Swiss institutions. And in the following time, in the rest of my presentation, even though my lab is mainly dedicated to designing this new multi-agent system, we started to work on how we can access scientific literature in an efficient manner for LLM exploration, Bayesian exploration. So this is the second part of my presentation. The project is called Perspicacity AI. It's about to be released. We had a paper accepted for a conference in the web semantic in the coming weeks. And you will all agree that the amount of research articles that are now produced is gigantic. It's hard to navigate, and it's hard for students, it's hard for researchers, especially in fields, and most fields are now interdisciplinary, that are requiring to have some notions or some expertise down from the instrumentation side to the data analysis part. So imagine in metabolomics, really needs to be familiar with mass spec, with analytical chemistry in the first place, good practices, Python, R, visualization, and so on. So it's very challenging to be a complete actor in modern science. Okay? And at the same time, we have now large language models that are available on our smartphones at any time, anywhere, and they are transforming how humans are interacting with knowledge. Okay? And so we have a discrepancy. We have a contrast between the challenge of dealing with vast scientific domain and these emerging tools that are extremely performant. However, and you might have tried some of these commercial solutions that are trying to connect generative AI with scientific literature. Most famous one would be Perplexity AI. They are using retrieval techniques to collect on the fly relevant documents, depending on the user questions, to try to propose an answer. However, those are, I mean, you might have observed that as well, they're relatively shallow because, in fact, they use abstract level information. They use some, let's say, not the deepest embedding of the content. The content is not available to this platform because it's protected. And also the knowledge base, or the set of literature documents, is not accessible to the user. And there are commercial solutions as well. So for us, when we started to look into that aspect, it was pretty frustrating. And so we started to think about what could be something closer to the scholar practice. Okay? So at the start of a PhD thesis, we would read the literature, collect all those documents, and organize them in a meaningful way. So this act of curating the scientific literature, it's at the base, at the foundation of the scientific research. Okay? And we are all doing that through a bibliographic manager like Zotero. Also, there are resources to search scientific articles on the web, including some that are representing these articles as knowledge graphs. And this is an extremely powerful way to find a new interaction in this vast scientific knowledge. More importantly, I think this is a key differentiator. As public institutions, we have rights to access some of the protected documents beyond the paywall. I mean, even without the proper access, it's possible to access the full text, right? So a commercial company cannot really leverage this common benefits as societies, as public institutions, we can empower. Okay. And finally, many useful information about good practices that are relevant for the training. It's actually now not just in the manuscript, it's part of the code repository. They would be on YouTube videos for conference or workshops. And all of that is essential if we want to access knowledge in an automated fashion. And so for this project, Perspicacity AI, that we started with some of the lab members, in particular Lucas, this project starts from your bibliography. So imagine Zotero. We would extract the bibliographic file. And beyond just your bibliographic curation, we can also collect research articles through recommendation and through the use of knowledge graphs resources. So one of them would be DBLP for informatics, OpenAlex knowledge graph also. And so we can combine your curated bibliography with some automatically collected, relevant, not always, there are caveats to this, articles to have a knowledge base that is relevant to your domain or your research. Okay? All that is collected, and we have a Python library that would go through each entry, download the content from the editor, the preprint, the repository, the YouTube transcript. All of that is collected and assembled into a knowledge base. We use a vector database for that. So we are embedding the documents into this vector database. And when this database is assembled, we can interconnect it with a large language model to perform what is called RAG. So RAG is a retrieval augmented generation. I will get back to this in a second, but essentially, when the question is asked, we are sorry, I had the French word in my mind. We are comparing the sentence with the documents in the knowledge base, and we are retrieving the most relevant documents and providing this automatically to the LLM to enrich the context and provide more precise answers. And more importantly, we can also, since we know which document was the most relevant, we can also track the source of the information. So we have a report with the citations along with the answer. So the system is designed in this manner. We will have the user of the Perspicacity AI system. The start is to have the bibliographic content. Either we will have only the curation from Zotero, or we can supplement with knowledge graph-based extraction. At this stage, we are using only keyword search. A alternative approach to this, one could use embedding of knowledge graphs, but sometimes these graphs are huge and they are not accessible easily. So keyword is the pragmatic way to this. And we produce this knowledge base of relevant documents. This knowledge base is connected to a retrieval augmented LLM system. And this system is more advanced than just typical RAG for the reason that we can use different retrieval modes. So I will detail more of this on the next slide. But more importantly, we are not restricted to the knowledge base that was created in the first place. We are also able to do external search on PubMed, for example, or Semantic Scholar that has some embeddings of documents, even though it's not comprehensive, to complement with relevant documents if they were not present in the first place. Okay. And as I said, the answer provided to the researcher is supplemented with the citations for all the information that was deemed relevant to provide the answer. And so we are reconciling what we would do essentially when we are doing literature survey, and that in a way that we can inspect, that we can verify, and not just like output LLM text. Okay, more details about the system. We are using three retrieval modes. I will start with the basic RAG. So I've described. We take the question of the user, and then we search in the knowledge base what are the most relevant documents. So articles, there are sometimes many pages. There's a need to chunk this document into smaller documents. So we are retrieving, I will not go into the detail, but we're retrieving like specific documents and the one before and after to have a window and not cutting down the context. And this is used to provide the answer in this basic mode. The advanced RAG is something you are probably not familiar with, is that we are taking the question and reformulating the questions in three different manners. Okay? Because the idea is that one formulation, it's a natural language processing, right? So one formulation would provide a set of documents, but a slight change in the vocabulary or the facet of the question will produce different documents. So we are decomposing the question in rephrased questions, collecting all the documents, combining them, and use this for the answer. In this project, we are also designing a new ranking function. I will not describe this, but the more documents that we have, the more it's challenging to identify which one are the key documents. And then the last mode, which is the slowest but the most advanced, is the profound mode. The profound mode starts by creating a plan to answer the question. So the system would get the question and say, okay, if I want to answer that, I need to collect information on that part of the question. Then I will continue with the second part, and so on. And at each step, the system can revise if this was sufficient or if there is a need to refine the plan. And it will call either basic RAG, advanced RAG, and so on. Might be supplemented with web search, and eventually provide, when completed, a summary report with the citations and so on. Okay. And this system for us, it's critical for many different tasks. At the start of a project, when looking at the literature for a paper, for example, for research proposal, of course. But I think for training, it's also very powerful because we can really navigate a complex literature corpus and start to verify and have a detailed examination of knowledge. And with knowledge, with large language models, this can be used in a very wrong manner, like without enriching the user, essentially. And for this project, Metabolic AI, we plan to use this to perform hypothesis generation in a semi-automated manner. So we would work on this plant species. We would have a knowledge base associated to it with the prior knowledge from the scientific literature and access this automatically to propose a hypothesis that would then be applied to the knowledge graph through a Sparkle query and the multi-agent system. So this is really the accessing scientific literature domain and interconnecting this with other AI systems. All right, so I presented our first prototype for multi-agent system on knowledge graphs for mass spectrometry, then the prototype that was used for this consortium, and finally our work on a system for scientific literature examination. And on a more ethical touch to this last presentation, I believe we are experimenting a real transformation in how scientific research is performed. And sometimes we see initiatives that are trying to replace scientists with AI, and this feels wrong, of course, but I think this is not the most powerful approach. The most powerful approach is really to think about amplification or augmentation of domain expertise, researchers, with these modern capabilities. And so there is a complementarity between what the community has as construct in terms of resources, in terms of methods, and the curation of the scientific literature, and these generative AI tools. At the same time, because we can access knowledge at the tip of a cell phone, a smartphone, it's also a challenge in ensuring that we are not impacting negatively the training of the next generation of researchers. So I think those tools have to be designed to make sure that good practice are included. And so for the last project, for example, we are keeping track of all the references and so on as researchers would do. So it's supporting this methodological framework. And something I haven't mentioned, I will mention this, for the multi-agent system, it's very flexible. So if someone wants to bring a new agent in, an agent for a MatchMS spectral matching, it can be done. Any computational method could be combined in a multi-agent system. So those could be really transformative for many different applications. And so finally, I want to thank my research group and also the collaborators at the INRIA, University of Genève, and funding agency also. So, and also all of you. Thank you very much. Oh, I wanted to do a demo, and I have no time for that now. So if you want to access either of the demo websites, you can use those links, and the password. This one is a contributor key, so make sure you use the right parameter, and this one is here. Do you have questions?
Yeah, we can have a few questions. You have like 4 minutes. Yeah.
So, well,
I don't know who.
I mean, usually the chair is choosing.
Justin.
So, thanks, Louis Felix, for this very nice talk. I remember the first steps of this metabobot in Germany, right, where we experimented a little bit with it and how it kind of provided some answers to indeed basic questions. And it's really nice to see how you developed it further now. So, what is your view on how can we keep this kind of AI systems indeed curated to the extent that they provide, let's say, the answers with good practices, as you indicated, because the literature is flooded, right? But luckily, mostly papers that follow, let's say, good practices, but not all. So how do we do that?
Okay, so your question is how do we select relevant scientific articles? Is that your question?
Yeah.
Okay.
In the end, maybe yeah.
I mean,
who's the judge of it, right?
Yeah. I mean, reproducibility, replicability of research is a very well-known issue, common to all domains. So selecting the relevant research articles is required to have some good foundation. Mm-hmm. And this is where scholar curation is essential. Automated creation of bibliographic content is very risky. So if we can leverage the domain expertise to ensure that we start from solid research articles, this is critical. At the same time, we are working on a project that tries to reproduce a research article from the start. It's too fresh to present. Essentially, the input is a research article, and we try to reproduce all the code automatically to see if it's reproducible. Mm-hmm. It can be replicated. So in the long term, such approach, I think, could be beneficial, because imagine we start to work on these plans. We are investing all these resources for many years, and we would have like few hundred papers. It would make sense to start to trigger automatic process to screen these articles, try to reproduce, and eventually flag the ones that are like suspicious or like not reproducible. So in the long run, I think there will be some automated way to kind of like triage the contents along with the curation, because this is irreversible.
Thanks.
We only have 1 minute. Was your question really quick?
No, you can come in.
Okay, then maybe we can switch to the next speaker.
Okay, thank you.
Thank you very much.
Is it this one? Working to you?
Yes. Thanks. Yeah, maybe I can do.
But wait, I think we downloaded it. Do you know how the presentation mode is started from Windows Edge?
Up there.
I'm not sure. Marnix, do you know from Windows Edge how you can start presentation? Oh, maybe just this?
Yeah, looks like it.
No.
Just need to move it to the other screen. Screen.
There was also a full page.
I think why not. I love Bungie.
And this. Oh, we got.
Okay, thanks. So, hello everybody. Thanks first, Elena, and also Justin and Marnix for the invitation. I think this is will be a great symposium and very interesting. My name is Pierre-Marie Allard. I work at the University of Fribourg and launched the Commons Lab in 2021. I want to share with you today the Earth Metabolome initiatives, and notably how we use semantic web technologies in the frame of this initiative. But first, a bit of background on why we are interested as humans, basically, in knowing more about chemodiversity. So first, for fundamental aspects, our colleague in ecology, they tell us that most of the species interaction, but also cross-domain interactions are underpinned by small molecules. Second thing is basically that this is the source of traditional medicine and also the source of inspiration of modern drug discovery. It's basically we use this chemodiversity to heal ourselves. And I think that this last part is basically what has fueled our envy to characterize these small molecules in the last century. And so in the last years, we have established with, as Louis-Félix said, so with Adriano, Egon, and others, we have established this Lotus resource, which is in the Wikidata knowledge graph, but you will hear more about this graph afterwards, which basically is an documents the occurrences of natural products across the tree of life. And once we have established this, basically you can draft this tree, and you see, so here the red branches, they basically tell you one molecule was found in this taxon, and you see that the tree is patchy. There are several reasons for this. I think that basically the fact that most of this research was done through bio-guided fractionation processes means that this takes a lot of time, a lot of money, and so explains why it's slow. And secondly, that there is a very strong publication bias. Basically, the journal in the field, they will accept to characterize or to share this research if the natural product is new, novel structure, or if it's bioactive. So it means that it will require a long time if we want to have a more in-depth view of this chemistry of life. But we are saved because we have mass specs and computers and a lot of people here developing nice computational metabolomic solutions. So what we can do in one day now on a typical platform is run hundreds of samples, and for each samples we get thousands of spectra, and with the nice tools we can get putative annotations, which is a problem. Yes, not an easy task. But and here I have three, I think, problems that I want to list with you. So first thing, we can discuss it afterwards. I'm happy to discuss it with you, but we have, despite the really nice aspects of mass spectrometry, notably sensitivity, structural determination, power, this data moves a lot. So M over Z, you can globally standardize this, but retention time intensity is moving a lot. So in this collection of 1,600 plant extracts that Louis-Félix mentioned, this is basically what you see when you get a PCOA. You see two big clusters. So this was acquired over months, so it's expected to have drift, but this was really nasty drift, no? So we tried to match which metadata was this, no? Is that above ground, below ground? Is that a plant organ or whatsoever? And actually it turns out that there was maintenance on the instrument at one point, and that you get these two batches. Second problem, the object that we study itself is plastic. Metabolome is moving, it's dynamic. So this slide was done to discuss with colleagues doing genomics and explain them the difference in metabolomics and genomics. So here I know a lot of people do genomics, so please be indulgent with me, but basically if you characterize biodiversity using genomics, you can take one species, you sequence it, and let's say, I put quote and quotes, that's it. You have the genome for this species. For the metabolome, it's different. No? You will profile the species, whether it's in stress conditions, whether it's a given organ, whether it's at different life stages, you will get different pictures. So this is illustrated by these layers that are similar to the layer that you showed. No? This colored layer would be one metabolome in one given condition. So we need to find a way to align them. But remember, the data is moving. Third problem, biodiversity is vanishing at alarming pace. This is true for all taxons, on all continents, and for multiple reasons. And there is one main responsible, and it's Homo sapiens. So this is why we wanted to launch this Earth Metabolome initiative. The idea is simple. It's to profile the metabolic content of all currently known species on the planet without taxonomical focus. That's plants, bacteria, whatsoever. I always say this when I present this slide. It's easy to make the slide, no? But it's obviously much more complicated to achieve. And we know that this will take decades. We know that we cannot do it in 1 or 2 labs in Switzerland. This needs to be a globally coordinated effort. And this is why I think we discussed today to try to find the possibilities to achieve such a type of project. So the idea basically for us is to connect to existing biodiversity characterization projects and to launch our own collection campaigns in order to establish a chemical extracts library. Then with mass spec and computers, we are able to acquire knowledge on this chemodiversity. And then, and I think that's the topic of the day, and I believe that this is one of the reasons why we think that it's possible to achieve such a project. We use semantic web technologies in order to align these heterogeneous metabolomics datasets across labs, across time, and across instruments. And it turns out that the structure that you make, which is a knowledge graph, is, as we Felix showed, very well suited to interrogate your dataset. But it turned out it's also very well suited to share this knowledge in public. So the resource would be this Earth Metabolome knowledge base, which is obviously an open resource for researchers, but also policymakers, industry, national parks, etc. And we believe that this can help, obviously, to advance fundamental aspects of life science, benefit human society in concrete ways. So, for example, drug discovery or finding novel source, sustainable source for food production. And in the end, protect biodiversity by what we call the establishment of molecular arguments for biodiversity conservation. So the idea basically is to try to build this virtuous cycle where you get knowledge from chemodiversity, share this knowledge, and try to use this knowledge in order to protect biodiversity and thus chemodiversity itself. So since this is a big project, we started with a pilot and we focused on botanical gardens. So why did we focus on these gardens? Because here, if you have ways to access chemodiversity, we identified this as a good in-between, let's say, what we want to tackle with the EMI, so wide ecosystem, where by definition diversity is maximal, and sampling obviously is complicated. And people, our colleagues working on model plants, where sampling is extremely easy, the plant is in the lab, but diversity is not that big. And just in between, you have botanical gardens. So here, we have a very decent diversity, actually. So in Fribourg, for example, we have a very small botanical garden, but you find these gardens in most of the big cities. And here, the diversity that we have is higher than what you can expect in the tropical Amazon, for example. So they are artificial biodiversity hotspots, if you want. And they are like tens of meters away from the lab. So that's what we started to do in the garden in Fribourg. You see here students collecting plants, taking pictures, putting them inside tubes, some going deep into the ground to collect them. This is basically where we stand now after 2 years of the project. So this is Botanical Gardens of Fribourg. We are around 60% complete. This is Alpine Botanical Garden in Champais, 2,000 plants collected. This is Botanical Garden of Neuchâtel, and this is very fresh data from basically last week, or last 2 weeks, from Kew Botanical Gardens, where Tito went with some of his PhD students and collected plants there. We used the citizen science platform called iNaturalist to document these collections. And so this is nice because on any smartphone, you basically capture the time of the collection, identity of the collector, take pictures of the plant. There is a computer vision model that allows you to identify the plant broadly. And then you also have community curation possible on these platforms. So even in botanical gardens, despite the labels, sometimes we have experts who tell us, yeah, you cannot identify at this stage, or no, you collected the wrong plant. So this is useful. There is an API, it means that we have programmatic access and we can fetch this data periodically. This is basically where we stand, so around 2,400 species and 4,000 observations. We've also done some BioBlitz, so we basically go in the field this was a team of 15 people, some experienced, some who didn't collect at all, in order to test the collection workflows. And this we've done in a vineyard, and we collected in one day 250 specimens, both plants, insects, fungi. So yeah, this picture part of the dried plant collection, for example, these are things that we are starting to do that we didn't do at the beginning. We start to add UUID, so this is a normally unique in the universe identifier, and you don't need a database for this, no? So you generate one and it will be unique. And we tag the plant with this. So this is very interesting because you see, for example, here, before we were just capturing the taxon. In the room, we are all, I guess, Homo sapiens, but we all have different names, no? So this is basically the name of this specimen, and it's useful because if you have different pine trees, then you can identify this one, and you can also come back to the specimen afterwards. So you see we collect a lot of data. So there's data regarding the ecosystems, data regarding the taxons and their organization, data regarding the samples that we track in the lab, data regarding what we generate on the mass spec platforms, and data regarding the database that we use for annotation. So the challenge is basically how do you make sense of this data? How do you organize them? You know? And so I guess you have the answer, well, need to do a knowledge graph. So what is a knowledge graph? Well, if you go on Wikipedia, which is a knowledge graph, you get this picture. So it tells you dogs, cows, they are animals, cows eat herbs. So this is the fundamental unit. It's called a triple: subject, predicate, object. Subject is the cow, predicate is eat, object is herb. To make it a real knowledge graph, actually, this needs normally to be identified with what is called a URI. You know? So think of it as like a URL. For example, in this case, you see eat maps to this relationship ontology or relation ontology, I don't remember. And if you go there, it defines what do we mean by eats. Adriana will talk you a bit more in detail about these things. Sorry, I'm going in the wrong sense. Yes. Okay, so we can also apply this to our objects of interest. In our case, so molecules, plants, and how they are connected. And this is actually exactly what we have been adding with the Lotus project. So we add this found in taxon link from data that is coming from literature, with the limitation that I've explained above regarding how this data is acquired. And this is what we want to do with the EMI, is basically knowing these limitations, we want to use distributed labs, collecting information using mass spec and computers to massively add this found in taxon link, bypassing the classical publication system. And I think that this is one of the big advantages of knowledge graph, basically. While we will be doing this, and we know this can take decades, well, other people, which we might not know, which we might know also, if they work in the same structure, so in the same knowledge graph structure, basically you can have other people documenting the fact that this molecule has a given antibiotic activity, that this plant is from a given country, that it has a given IUCN status, that this antibiotic has a given target, and that this target is the cause of these super serious public health problems. And so this path that you see in the graph is what we would like to call a molecular argument for biodiversity conservation. So yeah, this is not just concept. So Louis-Félix introduced ENPKG. This was, let's say, a first pilot where we tried to organize this metabolomics dataset of 1,600 plants in the form of a graph. What we get is 1,600 million statements. So these small subject, predicate, object. And basically, so here, yeah, I guess you will not see what is written on the node, but you have to trust me. This node, for example, is a node corresponding to a taxon. It's in Wikidata. This node over there corresponds to a molecule, which is in Campbell, so the chemical entities of biological interest. And tells us in this case that this molecule has a given biological activity. And so you can see the graph, basically, and the experimental data as a kind of metabolomics glue that is connecting these two data points. So you had two public data points, but they were not connected. And basically, that's the idea behind this graph. So regarding spectral information, we also include it in a detailed fashion because we kind of explode each of the spectra and take each of the content of the spectra. So the peaks, for example, and the losses, and each of the peaks and losses are encoded as a node within this graph. And this is quite interesting because it allows you to realize with this Sparkle query language, which was mentioned, it allows you to realize queries including the spectral information. So for example, in this case, we're looking for analog of this spectra, which was annotated with scopolamine, in another data set acquired on completely different conditions by just checking what was the spectra which has the maximal or same number of peaks and losses with this input query, let's say. And yes, okay, so I said that also these knowledge graph structure, they are useful to share information. So for example, you want to share information regarding the fact that this molecule was found in this taxon. And this bit of information can be shared with a small structure called a nano-publication. So this is what will be explored by Mishda in this Fermo or FermoNo project, where the idea is basically to try to be able from a tool that is organizing mass spec data to say, okay, I've made this small statement where this molecule was annotated to this sample, and I want to share this. So you just share this small graphlet, a part of the graph. Again, Adriano will give some information on this, I think. The thing is that, so another thing we would like to explore basically is share it also at a, let's say, at a bigger level, but not at the dataset level. So this would be at the analysis level. Each of the runs that you do on the mass spec with or without the metadata. And this, I think, well, it does not fit in a nano publication, but still, these small graphs, let's say, related to one analysis can be quite interesting because remember this picture that I showed you, so you would then have organize your sample metadata and the mass spec data, and you generate a graph. You do this for each of the layers, and given that you have the appropriate formats, well, you would be able to then generate this maximal common submetabolome. So we've worked in the last weeks on a small script. So this is very basic. It's just basically, so you have here MZML file, which you might be familiar with, which is an XML type format. So this is just parsing the file, getting these information, and serializing this to a turtle file. So here there's no metadata, no added information, and you now have a graph, and you can just stack your data like this. There are problems though. For example, we lose information here because we are doing binning, because we are selecting the peaks, but this allows you to realize this stacking basically. And the project that we are now working on, mostly in the lab, is basically building a portal, so the EMI data portal, which would have for objective, ideally, to help the user and the researchers passing from the field to the graph. So the idea is really to help and to be able to aggregate all the sample metadata information that you capture in the field, tracking the samples inside the lab, enriching the metabolomics datasets with the computational tools that the community is developing, and generating the graph. And so I have not that much things to show because for now it's just a huge code base. But I have two pictures. So it's a small crab and a big rock. Because the small crab is the logo of the Rust language, Rust programming language that we are using to build this EMI data portal. And the big rock is basically a symbol for a monolith. So a monolith, or a monorepo in this case, means that it's not distributed in different scripts, but it's one big code base which will have the aim to organize all these steps. And we use the Rust language because it's extremely fast, it's memory efficient, it's a strongly typed language, and it's checked at compile time, meaning that your code needs to work before it compiles and that you use it. And this works particularly well in the context of building a monorepo, or a monolithic, if you want, repo, because this for example, type checking will be active throughout all of your different stages. So you can have a look at the code source over there. And other thing that we do is trying to connect with other biodiversity characterization projects. So you might have heard of the Earth Biogenome project. They are clearly inspiring us. They've started in 2017, and the idea is to sequence life. So they focus on eukaryotes. And we want to discuss with them, notably how they organize data. And of course, I think, as we will see in this symposium, the idea is that knowledge graph can be, here I've shown just for metabolomics, but can be very useful to stack different omics together. But we notably also want to connect with them because we have identified that the pain point basically is going in the field, collecting the plant, identifying the plant, and putting this inside a tube. So when you do the efforts, ideally, well, you should do this, collect this, send this for people doing genomics, send this for people doing metabolomics, etc. Yeah, so I want to thank the people who are involved here, some of them. So there's Manu, notably, with whom we started brainstorming about this DBGI and EMI. So he's working in Neuchâtel now. You see him, he's an ecologist. So this is a rare picture of this guy in front of a mass spec. These two guys, Marco Visani and Luca Capelletti, he's the architect of this data portal, a rare picture of him in nature. So this is not completely true because the guy goes out in nature. Yeah, and the people in my lab, here's this Colin, the mass spec technician, doing the calibration. People who have joined or supported these initiatives, and also I want to finish on this slide. If you go on the website, basically there's a join us field. Just if you have any interest, we welcome anyone, I think, because this needs to be, again, globally coordinated. And anyone from bachelor level to PhD students, retired PI, as long as you have interest, I think this is the idea, to welcome everybody. And so, yeah, thanks again, and I'm happy to take questions now or after during the day.
Well, thanks, Emily. So you described it very, of course, enthusiastic how you are building like more and more knowledge. What do you think are the challenges that you're facing now in kind of expanding that further, this knowledge base?
Yeah, well, I think when we can discuss it along the day, but I believe that we do have the technologies and to do this. I mean, semantic web technologies, they emerged in the 90s, so just after creation of the web. Mm-hmm. I don't think that it is that complicated, and if we require some tool, we will be able to make them. No? The most challenging aspects, I think, is really, being aligned, let's say, as a community on, for example, sharing vocabulary. So we know that there are ontologies, right? Okay, but what happens when someone comes and say, okay, what about this term? What do I do about this? I don't find it there. So the first thing would be to have really efficient method, let's say, to say, okay, this domain expert has this concept. Okay, you look at all the ontologies, could be this, this, and that. And then also, I don't know, a kind of forum or something that is public that would allow people to say, okay, no, you should use this term in this ontology, or no, yes, this makes sense, we should build yet another ontology.
So you're arguing it's alignment of data that is close to be solved, but it's alignment of communities and how we talk to each other that is the biggest bottleneck now. Yeah,
well, I wouldn't say it's completely solved, but I think it's not the most challenging aspect, yes. And I think that, yes, sharing common vocabulary is the same, no?
Thanks.
Yes?
This might be a bit more general knowledge base or a question. So how do you deal with uncertainty? If you put it sort of in knowledge graph, then it becomes knowledge. But very often you're not 100% sure. There could sort of
exactly
alternative explanations as well. So do you include that in the knowledge graph, or do you ignore that?
That is, I think, also could have added this to the challenges. I think one of the very important topics to address, I think it's linked to also Felicia's questions regarding Denis, sorry, questions regarding metabolite annotations. We know that when we do metabolite annotation with mass spec, well, almost no one can tell you, yes, it's this molecule, I'm 100% sure. So we need to encode this uncertainty, yes. And again, I think Adriano will address particularly this point, notably in the form of, there are different ways to do this, but basically the idea is to document the predicate, for example, with another set of triples that can tell you, okay, I've done this using this tool, this version, with this given score. So then you know, and you are able to change basically between, okay, I have isolated this molecule and this is backed up by this paper and NMR, versus I've done this using this metabolomics solution. But this is not, I think, yeah, completely solved, and, but very important.
Maybe one last question?
Yeah.
Like when you're now talking about biodiversity conservation, what do you think are the challenges regarding, for example, legislation of the use of biodiversity and derived data? For example, like making publicly available genomes from like very endangered plants or fungi or bacteria, and like Nagoya, for example, like how do you cope with these things?
Okay, so Justin, I could also have added this to the challenges. I think this is an important, very important one, a crucial one. We a bit naively started also with botanical gardens, saying, okay, it's secure, we go for botanical gardens. And you can discuss with Tito, who was in Botanical Gardens of Kew some weeks ago. No, it doesn't work like this. In gardens, they have CITES plants, they have protected plants. Not all of them, actually, even in the best gardens, let's say, are Nagoya compatible. So all these things need to be discussed. And when we will be doing collection, and I think this needs to be clearly addressed with the focal point of each of the countries. Notably because we will basically share openly data regarding the chemistry. And as you know, well, chemistry, it's immediately linked to potential application, patentability, etc., etc. So these are fields where we would clearly like to be more in touch with lawyers and specialists of these types of laws, because one of the things that we want to do absolutely is share this data under CC0 license when it's produced, as soon as it's produced. There is no embargo, there is no logging possibility or whatsoever, so an industry could not tell you, for example, okay, I want you to provide these type of taxon, but we will get the data for us, and even then afterwards you release it as public. So we think that this can be a way basically to cut under patents. I mean, the stuff is public, it's there. Yeah, I need to get in touch with people who know or more knowledgeable about these aspects. If you have ideas or contact, very happy to take that.
Well, thank you. Thank you again for the speakers of this first part. And we have a coffee break of half an hour, 1 floor downstairs. Say something to. Okay, welcome back. Let's start the second part of the session. Welcome,
okay.
Adriano.
Thank you. Let's see. It's working? Yes, good. So, as said, I will talk a bit more about a specific knowledge base that is Wikidata, that we use to document also part of the initiatives you heard about. And hopefully you will also see some technicalities on how it works behind the scenes and get used to knowledge graphs. So first, I think this slide is probably something you can remember. This system was introduced by Tim Berners-Lee when he launched the web, and basically it's a star system where if you put something. Nice. If you put something under an open license on the web, well, that's the first start. Of course, you need to put it under open license, but you get a first start, which is good. And most of what we publish is not even 1 star, 0 stars. So then you have something that is machine readable, that's 2 stars, machine readable and in an open format 3 stars. If you start describing things a bit better with prefixes, URI and so on, you get the 4th star. And if you link your data to others, you get 5 stars. So you could say, okay, 5 stars is the best. My view is Wikidata is 6 stars because it's collaborative. So it's not only linked to others, but also edited by others. And this brings really interesting results. So how does it link to what we mentioned? The slide is adapted from Pierre-Marie. And basically, what all of us, I think, roughly study is a taxon coming from Earth. It might be human, plants, whatever type of organ. You acquire some data. In our case, metabolomics data, so spectra, it could be genomics, it could be sequencing, whatever. But in the end, you obtain a small part of data. And usually this is roughly easy. But then connecting back this data to real knowledge is the challenge, and we aim to do it with knowledge graphs. So, in our view, you start big. You use whatever type of microscope, a machine, to reduce the dimensionality of your data. In our case, mass specs, and then you need what we call a macroscope to gain context back again. And so, in our view, you generate data, eventually you convert it into open format. If you share it, 3 stars, good. But then you need a lot of dedicated tools to process it, and going back again to biological meaning, biological function, linking it to activities or pathways, and we'll have 2 beautiful talks about wiki pathways later. And this is our view on how you go. First, small, and then large again. And actually, we were reading a bit of the old textbooks from the 80s centuries, and it was already there. So of course, you need to call everything omics, so we called it taxonomics. And basically, what this guy that was a Swiss botanist said is that plant taxonomy would be the most useful guide for basically medicinal plants research. And so we thought a bit about it, and yes, makes totally sense. You have the morphological trait, the taxonomy on the one side, the genomics in between, and then somehow the end product, the phenotype, the metabolite. And back during my thesis, I did this taxonomically informed metabolite annotation, where basically you take advantage of the taxonomical context of what you're studying to gain confidence into your metabolite annotations. And when we did it, it was working, but with closed tools. So this is why we then decided to switch the situation from exactly those 2 to 3 stars situation to 6 stars. So basically what we did was taking all the available natural product data, we did some semantic elevation. So we converted it into linked open data, and we uploaded it to Wikidata. We're not the first ones to do something similar. So there are some other initiatives that were already done from Andra Wagemeister and also Ikan that was here. And there are more and more examples of Wikidata being used as a knowledge base for life sciences in general or other sciences. So I will try to, let's say, make you less scared of triples, and eventually you will see that you can also become a Sparkle master without too much issues, and also with the aid of chatbots, you can make it. So as said, subject, predicate, object, that's the basis. If you think about Wikidata, Wikidata is an instance of a knowledge base. And this is really how all these triples look. Maybe if you're a bit more into the details, you have a prefix, an identifier, and that's what you need. So if you start going broader, Wikidata, one of the main advantages, it's multilingual. So if you contribute to Wikidata, you're not contributing only to an English-centric knowledge, but to something that can really be reused in many, many, many other languages, because we still have a huge bias with English published knowledge. And as Pierre-Marie said, so this is the only thing I've found to link basically WUR and Wikidata. So it's not a biodiversity conservation argument, but with the links you have, you can link that YouTube influencer that is born in Wageningen to Wikidata. So you can have also a short path on knowledge graphs to link whatever you want. And what we wanted to link were chemicals and taxa. So if you start with chemicals, the most basic query you could use to do it, and I want to try to do it live. For all the queries, you will have them on the slide. I try to document those because usually when you start trying to query, if you have some example, it's quite nice. So Wikidata has a query service I can run. You can simply have the query here, you click on run, it runs, you get the results, it's quite fast. And the thing I wanted to show, because this is like one of the most precious tricks you will hear about, there is auto-completion. So if you do control space, you can start just typing what you think about. So in that case, chemical compound. Okay, it's just chemical compound. And then you have the ID, so you don't need to know the ID. Sometimes it feels like black magic, oh, they all know the IDs by heart, I don't. So you can simply start typing and it works. So I'll just come back here. And so today, 66 results. So you might say, okay, wow, 66 compounds, that's not a lot. Why would I use Wikidata while PubChem has much, much more? Well, there is a small detail, and I think this relates to ontologies alignment that was already mentioned before. There was a rework on Wikidata, and chemical compounds are not instances of chemical compounds anymore, but types of chemical entities. And if you don't know it, well, of course, you end up with 66 results instead of 1.3 million. So there is a lot of guidance on how things are modeled per project. So this is the link to the chemistry project, where Egon here contributed a lot. We also have a preprint where we explain it, a bit how you access chemical data on Wikidata through Scolia. And you will have all the links. The talk is quite short, so I cannot mention everything. But then you can do the same on the taxa. So the taxa, there are many more, almost 4 million. And if you then try to look for IDs to link them to your NCBI sequences, Open Tree of Life IDs, or things like that, you can start making some deeper queries. And I just try to highlight some verbs that you can use in Sparkle. So you can use optional, so the field is not necessarily needed, or minus, so that the items that have an Open Tree of Life ID are not shown. And here, this is another interface, another endpoint to Wikidata that uses Clever, and this is also something super nice with linked open data. Basically, you can access it from many different ways. This is another way where you do not have some limitations, you have some others, but just to show you, basically Wikidata, you can access the data in many different ways, formats, and then you can download as JSON, CSV, or. And as you can see, for example, some taxa have an NCBI ID, but not the GBIF ID, and so on and so forth. But yes, I said we're interested into the links between chemicals and taxa, not only the two objects without their links. So this would be a query where basically you're looking for things that are chemical entities and how they're related in the form of the predicate to things that are taxa. And if you look at it, well, there are 500,000 roughly that use this predicate that is basically found in taxon. So this is what we documented with Lotus, molecule found in taxon. And you can also see there are some other properties, for example, taxa known to produce chemicals or other properties, but roughly the one you need to remember is found in taxa. And this is the backbone of what we uploaded from Lotus. So you do not have only the compound taxon pairs, but also the reference. So if you look at it, you have, again, compound, taxon, and from which DOI reference it was inferred. And here you have almost 700,000 of these triples. So usually the first question I have is, okay, but I don't want 700,000 pairs. I'm just interested into Piper nigrum. Please give me the list only of Piper nigrum. So you can also do this. Again, if you use the auto-completion, you can just start typing your species, and it will give you the list of the molecules that were reported in Piper nigrum. So here, in that case, 400. And probably you will not be impressed. You will say, oh, 400 molecules, I could do it on my own. I can query a database, and yeah, 400, not impressed. But what if you go to Piperaceae species, Piperaceae family? So there, okay, still almost 2,000. Maybe some of you might be brave enough to do the literature search of Piperaceae. But if you go to magnolids, so a clade of plants, well, 10,000 and still taking a second. And this is just showing that you can go up in the tree without too many limitations. And this you could not do using one-star PDFs, I think. So you can just imagine the amount of PDFs required to generate those 10,000 lines. It's quite huge. And the nice thing Pierre-Marie also said, we started documenting this molecule found in taxon links, but on Wikidata you have everything, basically. So YouTube influencer, footballers, and people that are interested into taste. So some people started saying, okay, this molecule is known to be bitter, or is known to interact with that bitterness receptor too. And so you can also query for this link to taste, and you can see that among molecules found in magnolids, well, 120 almost are known to be bitter. And this we did not upload the data, we're just benefiting from other people's work. Maybe relevant for some people here, there was that review, I think in 2021, of natural products and their bioactivities in actinomycetes. And as a small test, we wanted to see, okay, can we reproduce that table 1, where they're listing organism, compound, bioactivity. So we did it. We ended up with 571 results. I will not tell you how long the table 1 is, but you can basically reproduce your whole review with one query. So it's quite nice. And this also leads me to the next one, that is the link to other omics. So I was heavily talking about small molecules, metabolomics, but you also have people that started putting proteins there, protein interaction, genomes. And you can ask, for example, for Pseudomonas, how many taxa have how many links to metabolites, genes, proteins. And I think what's quite interesting is that if you look at the overlap, it's almost none. Because the metabolites are described in that Pseudomonas, while the genome was heavily studied for that specific Pseudomonas strain and some others. So there you can start seeing the interest of filling the gaps in between the different type of omics. There is another super cool aspect you'll benefit from on Wikidata, and this is the 6-star. It's community curation. So especially for structural elucidation, we know sometimes even in literature people report the wrong compound. And if you go on Wikidata, you have some triples explaining, oh, okay, this was actually the reported molecule, but it was wrong. And there is another paper that said, oh, this molecule was not the right one, there is the new one, and you directly benefit from it. So if today you upload your data and to the best of your knowledge, someone might be able to say, hey, now it got even better, there is a refinement, so you just have this without having to host your own database and someone sending you an email asking, oh, this is not true anymore. They can do it on their own. Another thing that is super cool, you have community gadgets to improve your experience on Wikidata. So one of them is made by one of our speakers here, and you can display the structure. So maybe you can read smiles, it's quite hard, but seeing the structural depiction might help a lot. So just by enabling this small gadget, you have depiction on Wikidata, which is super nice. You also have some other display where you can see, okay, this guy I would never trust if he uploads something on Wikidata, or okay, it's someone knowledgeable in the field, so maybe it's a good statement. Yes, and there are plenty of other enhancements you can use. And the whole reason why knowledge graphs and semantic web technology is interesting is federation. So federation means you're asking different graphs for data. And here, for example, I highlighted these lines. So basically, you're just querying a smaller subgraph in Wikidata about references. And this is something rather technical, so the graphs are split in Wikidata, and you can access them from the same query, and you might say, okay, it's quite nice. If you just, there is a small visual, basically, of the query, what it does. So this tool was developed by the Swiss Institute of Bioinformatics, and basically you can see what the query does, and it asks another subgraph here about that precise relationship. And this is, let's say, a Wikidata only graph, but you could also, okay. Yes. You can do a federated query on, let's say, Wikidata only, or you can start going outside of Wikidata. And this is honestly one of the best resources you will find for small molecules, which is the integrated small molecules database that is developed in Prague. And there, well, you can federate also your query with Wikidata, and they have additional features on that database. So here, for example, you have a substructure search. And if I click, well, you have compounds that match a given substructure scaffold. So you can use that part also in Wikidata and query for specific substructures. And honestly, just try them. There are so many things. And Pierre-Marie mentioned mass spectrometry search. There is also something like this, and you can search for mass spectra. So here you are looking for those peaks. And if a molecule has that given mass spectrum, it might take some seconds. Hopefully, yes. And so you can see molecules linked to the spectrum that was used to do the matching and the score. So you can start really leveraging totally different types of information just because of that interoperable format. So this one I showed you with the mass spectral search. And maybe the absolute coolest one is the one with RIA. So here you can see you're really having different prefixes, some of them of bioinformatics, chemoinformatics, and then you use the RIA knowledge graph to query for compounds that are known to be parts of some reactions, and link the spectra to those. So you're doing something absolutely crazy. You would have needed, I don't know, different types of CSV databases, external tools to do the matching. You can just write a Spark query, and you're querying things part of a reaction that match a given spectrum. I find this amazing, but maybe I'm a bit biased. Yes. And again, this was work done in Prague by Jakub Galgonek, and it's really cool that we have such resources available. So maybe just to wrap things up, during my PhD, I was interested into bitter compounds and the plants they were found in to elucidate the bitterness of vermouth. And this is why I came up with Lotus. And then we realized, oh, okay, Lotus can actually be used for NMR-based dereplication and mass spectrometry-based dereplication. And after some time, it was reused by many, many other technologies. We also saw the experimental knowledge graph, the DBGI, and so on. And I think what really matters is eventually you. So if you think about it, each one of us is interested into small links, a small subpart eventually. But if you make them available in such a format, it will benefit communities you didn't even think about. Because they will be like, oh, this is interesting. Butterflies' phylogeny limits to what they eat and the molecules that were found in them. So, yes, almost no limits. Sky is the limit. As Pierre-Marie and Louis-Félix already said, we're starting to make this graph also a bit fuzzier, so adding links that are experimental ones, not necessarily real ones, but with associated probabilities. Like for your question, we call them non-accepted triples. So those are things we assume that might be correct, but for now, we do not have the final evidence they are. Acknowledgement, so the whole team behind Lotus, Swiss National Fund, my lab, and in case I did put some useful links in the end, they will be available, and you also have all the references. Happy to take your questions.
So also because of my fault, we were a bit delayed after the break, but maybe like 1-2 questions we can take. Mitya?
Yeah, thanks for a very nice talk. There's 2 questions. The first one, a more practical one. Are you going to make this available? The slides as a note or something?
Oh, so the slides are for now on my website. I'm happy to share them by link.
So that would be then adafede.github.io.
Yeah, exactly. This one, and you can find the slides. Yeah.
And the second, the actual question was, so it's very inspiring, but perhaps you can also mention briefly the notability guidelines of Wikidata. I think that's very relevant to this audience.
Okay, so basically, yes, Wikidata, and this is the reason why we did build the experimental natural product knowledge graph. Wikidata has some notability criteria, and you cannot simply upload your mass spec experiment there or your, I don't know, your specific sample tube. So it's the backend behind Wikipedia, and if you create, let's say, a new molecule, it needs to be documented somewhere. It cannot just be on your lab book. So external identifiers, or you need something notable according to Wikidata, to Wikipedia media rules. And if you think you will have plenty of objects of your own interest, then start building your own knowledge graph that will then federate with Wikidata using exactly those mechanisms. But yeah, eventually I would say put as much as you can on Wikidata, because we're also paid by public money, so giving back to public some useful information to feed Wikipedia articles is good, but eventually not each and every bit of your experiments that you can then accommodate as nano-publications or on other more scientifically related knowledge.
What happens then to interests that don't feel the notability route?
Good question.
Deleted or?
They're not. So it depends. You will probably get a warning that you are not following the guidelines. First time it's a warning, then your account might be blocked if you use it. But I would not go into that direction anyway. So usually it's really rare that something gets deleted. It's just for edge cases.
Thank you again, Adriano.
I don't think I need a microphone.
But maybe it's important for the recording, right?
Yeah.
It is?
Oh,
yeah.
Oh, yeah, then I will.
It's okay.
Yeah. I didn't wear a shirt to put on
That's
such
okay.
a microphone.
Maybe almost.
I'm trying again. Yeah, is it okay like this?
Yes,
Okay. Okay.
and this is the point.
And then. Okay, cool. So yeah, I have a very loud voice, but the microphone is apparently needed for the recording. So I'm Denise. I'm currently a postdoc in the Translational Genomics department in Maastricht University. Very happy to be here. Part of this presentation I have given at the PhD defense of Andra Waagmeester, who was also previously mentioned, who used to be in our group and is now at the VU in Amsterdam. And there is some overlap with some slides. So some of you stole my slides. I don't know how that happened. But for me, the added value of using linked open data is that you actually structure the data and you can interlink it with other data. I will have some examples of knowledge bases that do not allow for that, such as Neo4j. I did use them in my project, but it's not the same thing. You can make the data that's in there useful through these semantic queries, and it builds on standard technologies that are already out there that we use for the internet. And it actually gives you more than just a website that humans can understand, but it also makes sure that the information is computer readable. Now, this slide you've already seen, but this one you did not, I think. So this is a visualization of how Wikidata is linked in the landscape of knowledge graphs. This is a really old visualization. It's just on the Wikipedia page. But even then, there was already a really big link there with Wikidata to a lot of other resources. So what I would like to show you, and I really appreciate the previous presentation with all the Sparkle queries. I will not show Sparkle queries, but the way I use the Sparkle queries to actually interact with metabolomics data. And then I will first start with an example of metabolomics pathway analysis. So the problem is with metabolomics data is often the size, right? We have some data points that are isolated. And if you compare to transcriptomics data, we are just not meeting what they have in terms of size. They have 10,000 data points or more. I'm very lucky if I have 500 data points, 100. That's just a reality, and that's not because of LCMS, but all the techniques do that, unfortunately. But what we want to do is we want to understand these data points. So we want to cluster it together. Say, okay, these are found in higher concentrations, this one in lower concentrations. And for genes, then people really look at functional groups. What are these things doing? But then we also want to know, how are these things organized together? So can we actually link the function to, for example, a pathway? And then the next step would be to integrate these pathways in a network, because we all know that the product of one pathway is probably the substrate of another pathway, just no one decided to draw that link. So what I looked for in my PhD is, can we actually use linked data approaches for omics analysis? And I'm really going from the transcriptome to the metabolome, maybe also integrating the fluxome. But that was a lot. So then I decided, well, then let's focus on the metabolome and the fluxome and yeah, the genes, that's for someone else to worry about. So if you have a metabolomics data set, and I just picked 4 random examples, 2 are endogenous metabolites, but there are also some exogenous metabolites, and you also find those in human samples. And we also find a lot of plant metabolites in human samples. Then I want to do pathway analysis, so I picked 1 random pathway, and I find 1 hit. Let's try to do some statistics on that. It's not going to work. So we have a lot of data sparseness. The data coming in is of a small amount. Then you link it to something, but there's also not a lot to link it to. We have a lot of identifier mapping issues. And also the coverage between different part of databases is different. So you have databases really focusing on plant metabolomics, but you also have some for human metabolomics. And the coverage is just not the same. So you often need to integrate information from multiple databases to actually understand your metabolomics data set. Now this is based on work that we did not publish yet, but we're aiming to do so, where we looked at different data sets. They had transcriptomics profiles for both microbiome and the host itself, and they had metabolomics data. And then we said, okay, what if we want to combine that data? Because we have a lot of transcriptomics, a little bit of metabolomics, maybe together that will increase the power. It did not. Why? Because the highest number of significantly changed metabolites in the data sets that we could link to a pathway is 8. Yeah, I can do statistics on it. I tried. I added a p-value. Is it reliable? Probably not. So the problem, what we wanted to in the end tackle, integrate transcriptomics and metabolomics data together, was just not going to happen. Because that one data set that I have with 8 metabolites was not significantly changed for the transcriptomics data. And then I only went down to, okay, maybe there are 4 significantly changed metabolites. Is there any link to the transcriptomics data? If there was, it was not significant for transcriptomics data, or the pathway was really large. So then I have 1 metabolite here and 1 over there. How am I going to understand what the relationship between these 2 is if just the distance between them is really large? And just, yeah, the little coverage of metabolites in general. So that's really hindering at the moment doing some form of multiomics. What we then did is we said, okay, but wait a minute. We have 2 different diseases, right? So this is a human data set. I normally don't work a lot with plant metabolomics data sets. Ulcerative colitis and Crohn's disease. And there was 1 pathway that they both had in common. The sphingolipid metabolism pathway. We said, okay, let's just pick that one because, you know, at least there is something there that's overlapping. And we visualized it together with the transcriptomics data, and then we had 2 transcripts in that data set that we could actually visualize on the pathway. So there are still so many challenges that we need to tackle before we can actually integrate this data to understand the data, right? It's not only for having a nice visualization. And the identifier mapping issue was really prominent for this case, so it had a lot of lipid metabolites in them, and the data set was annotated with outdated identifiers. And you see that a lot as well in data sets. That a database had an ID, then they released the data set, the ID changed, and no one bothers to fix that. And then I want to do some downstream analysis, it does not work. So we are working on something for that. The manuscript is in preparation, and I really need to find some time to finish that. But it's already available in GitHub. We have a Shiny app for people that do not want to program. But if you want to program, also all the data is there in the Sesame format. And you can just integrate it in your own tool. So the next would be for me is to show you a metabolic interaction graph database. So that's not exactly the same as just connecting the metabolites, right? So if you have that connection, you have this network, at some point I also want to understand how fast are these reactions going? What is the weight in this knowledge graph? If I put in more of one, will it also be produced something more downstream? So for that, we built this pipeline and we integrated 3 different databases because I said previously, right, the coverage is just not good enough to rely on one database. We're very happy that Reactome shares their data open source CC0 and that we can integrate it in wiki pathways. And then with a collaborative project of the lipid maps team, the pathways they had were also now integrated into wiki pathways. And what we have in there as well is that we have a curated collection. So the word wiki for some people means no one checks it. We do check it. And also in this pipeline, I have 2 types of quality control. But there is also data from models people are still working on. So we differentiate between that, and the releases of the data that we do, the monthly releases, are only that curated collection, so that actually someone had a look and it says, yes, this is good enough to release to the public. But in the end, I had 4 different types of input for my data that all automatically goes to RDF because of the nice code that Egon wrote for that. And then the thing I was challenged with is that I wanted to write one query, and then I got timeouts, and I was missing data. I was like, okay, I'm just going to split it in 3 queries. That's at some point the problem that you're running into if you're aggregating a lot of data, that you just get timeouts. So I split it in 3 queries, and then I wrote some code to put it back together, because that's what I needed in the end. I will go into the graph model in a little bit more detail later. And then we stored that in Neo4j. So Neo4j is a knowledge graph, but doesn't have semantic encoding for the data in there. The reason we use Neo4j, because when I started this project, it had really nice algorithms to actually connect different data together through shortest path analysis, which is what I needed to not have a big hairball in the end with a lot of data I cannot interpret. And there was already an existing app, Neo4j, and then we extended upon that to do the visualization in Cytoscape. That's used really a lot in our field, and it has lots of nice visualization options that the free version of Neo4j does not have. And you can automate it with an API. I just quickly had a look at my data set, and sorry, they're all LCMS or GCMS. So the data that I use is probably not as good as I want, but just to see, is it working? Can I link a data set to the network that I have? So this I already explained, and the details are in this publication that's available in Digital Discovery. And then this graph model, this was very problematic. I had to redesign that 4, 5, 6 times, I don't know how often, just because of the downstream applications that I also wanted to allow. So what doesn't work in networks is edges on edges. Does not work. You need to put a little note in between. And this is problematic because in chemistry, we often have that, It's not always A becomes B. It's A plus C becomes D plus E with this enzyme catalyzing the reaction. And that is something that these knowledge graphs are struggling with, and a lot of the models that we use are also struggling with. So what I decided is to say, okay, I have a substrate, and then through some reaction, it becomes a product, and it's catalyzed by a protein. Now I also have some side metabolites that are there in a lot of reactions, water, just a random proton, ATP, it's there everywhere. But if I use that for my shortest path algorithm, just everything is connected through ATP. And that's not my main goal. What we also added in there were mappings from different databases. So we based the main reactions on Wikidata because we can curate that ourselves. But most of the metabolomics datasets out there do not have Wikidata as the initial identifier, right? They have CABI or HNDB or something else. So we added those as well. And then what we were left with in the end were over 16,000 nodes and 34,000 edges. And the reason why we have so many edges is, one, because I add this reaction node in between. So you actually double the amount of edges that you have. But the other thing is that we wanted to provide provenance. So where is the data coming from? So we have one interaction that says, well, this is just everything all together from all these databases. We have one for Reactome, one for Wikipathways, one for lipid maps, and then one for the ones that are actually curated. Yes or no? So you can filter, and that's a nice thing about Neo4j. You can filter for the shortest path algorithm on the label you put on the edge, which is slightly more difficult to do that with Sparkle. And because we actually added also the identifier mappings, we resolved that issue. We also tried to resolve the different coverage and pathway databases just by adding 3 databases. It still doesn't cover everything, but it's a start. And also try to resolve the low amount of data, because if you integrate all the pathways together in one network, that product that was previously not connected to a substrate is now connected, because it's the same ID. And then you can continue from one pathway to the next one. Now then, this is the hairball that I create with my shortest path algorithm. And if you've seen these type of network analysis before, this is actually a doable hairball to look at in a bit more detail. So if I zoom in on, for you it's the top right, what we looked at in this data set, it's not my data set, it's from a group in France, and they put that on the metabolites platform. And they looked at which type of metabolites are related to aging. Now, I have to say this data set was done with volunteers from the university, and apparently caffeine metabolism significantly changed in people from university. Who knew? But what I also saw is they, in the end, wanted to make a model to say, okay, if I see these metabolites, can we kind of predict your age? But these 4 diamond shapes, you cannot model those as individual variables. They're so close together, they're actually influencing each other, right? If you drink caffeine, that's actually metabolized into these 3 products. So putting that as individual variables in your model will probably not give you a lot better results because they're just related. They're correlated to each other. Then if I zoom in on the right-hand side, what we saw there was that there were 3 metabolites found in a lot lower concentrations when people age. And then I saw, okay, actually in the middle between them, there's oxytryptan and serotonin. And then I look for literature. Oh, oxytryptan is actually known to decline with age. Now, the link to serotonin is not there. Because if that's not in the database, you will not find it. But we can link this information back to literature. So if you're interested to know more about metabolomics data analysis, we actually have a meeting coming up in Leiden, where a lot of people that are also here are going to be teachers in. So you can sign up there and join us in Leiden for this next conference. And then the next part, so the one focus was to develop tools, but the other one was also to apply it. So I was very lucky that the Maastricht University Medical Center, their clinical genetics department, was willing to collaborate with me on rare diseases. And because of my focus on metabolism, we looked at inherited metabolic diseases. And to give you a bit of an idea, because I think a lot of you work more with the plant realm, what we're actually suffering from here is that the substrate becoming a product, that reaction is not happening anymore because the enzyme catalyzing that is not functioning or has partial functionality. And that actually leads to, well, there's just less product because it's not created, and that can create deficiencies. So also some substrates are accumulating and can be toxic. And both of these then trigger some downstream cascades. Now, in the current diagnostic process, and what happens a lot with these cases, these rare metabolic diseases, for those of you that have looked at the 3VM series request of last year, MetaKits, for the Dutch people here, that was actually working on trying to get funding to study these diseases. Because a lot of these diseases occur when children are very young, and a lot of them are lethal. And there's not a lot of treatment available for these diseases. So your child is born, and within 1 or 2 weeks, they start to deteriorate. They're not really thriving. You go to the hospital, and at some point they say, okay, maybe it's a metabolic disease. You go to a metabolic pediatrician. They write down some symptoms that they see. They take some samples, and then a lot of other people are involved to, in the end, come up with an interpretation of what could be potentially causing this failure to thrive. And actually, the hidden layer in that is the database curators or modelers. I did add them in here because they're very important to actually make sure that we can link the sample to the interpretation. And what they do in Maastricht together with Nijmegen is metabolomics analysis, whole exome sequencing, also some RNA and enzyme assays. But the metabolites and whole exome sequencing are the ones giving the hint, okay, this is probably the enzyme not working. And then they do the follow-up experiments with the other ones. But the problem with these diseases, besides that they're rare, is also that the patient population is very heterogeneous. So you can have 2 people with the same mutation that have different symptoms. You can also have people with the same symptoms that have different mutations, even in different genes or proteins. So that makes the analysis really complicated. And even if the variant is known and the biomarker is known, it's still difficult to have a diagnosis for these people fast enough so that if there is a treatment, we can start with that soon. And metabolomics actually works for both cases, right? So if the variant is not known, you can still use metabolomics. Whole exome sequencing will then not always help you. But if the variant is unknown and the biomarkers are also unknown, it's really difficult to come up with a diagnosis. And what we wanted to do in this project is try to visualize the metabolomics data on a pathway level to see, okay, maybe this enzyme is not working. We do not measure all the metabolites in the pathways, but like 1 upstream and then 3 or 4 downstream. It's maybe this enzyme, so to try to trace that back. And for that, what we actually did is we extended the model we have in Wikipathways. So we used GPML, that was also previously mentioned. And we added these little pink boxes. I don't know why I decided for pink. That was just a color we were not using at the moment.
And we say, okay, this is the substrate, this is the product, this is the enzyme, and this is the disease linked to that reaction. Now, if you look at that from the RDF perspective, and this is the harmonized form of all the data we have in Wiki Pathways, this label that I used is not available. There's actually a level in between, so that's when everything from GPMLs is transported into an RDF. That looks like this. So that's already a lot more complicated. We can make the link of that disease label to the substrate product and the enzyme, but that's like 10 steps more than what I would like to do. So from that, we found out, we did a lot of this with community, so with students that needed to get ECTS, and then they find me and I make a project for them and then they work on that. That we actually need clear modeling guidelines, because the easy thing is substrate, product, enzyme, but we know a lot of chemical reactions are not like that. We also need to make several updates to our code to make sure that this information does get harmonized into the Wikipathways RDF and can be queried easy. And yeah, some biology is just really difficult to capture. And then putting that in that easy scheme does not work. We have a preprint out there in bioRxiv that is explaining this also with some example Sparkle queries, because the data is available and you can load it in a local graph store as well. And then to connect it actually to the disease data that we have from the hospital, this is the pipeline that we developed for that. And as you see, there's quite some RDF involved in there, some identifier mapper from BridgeDB. The problem I had was this IEM base, where the theoretical biomarker data is loaded in, no API, no Sparkle endpoint. Basically, you can go to the website and find something there by typing something yourself. That's how it worked. Then they had a nice table with theoretical data, with new and old ancient BIDs. Everything was a kind of a mix. And then to say if the metabolite was significantly found in higher or lower concentrations, they use arrows pointing up and down. Try to convert that to something a machine understands. Well, I found another student, and she made an Excel sheet and put everything nicely in there. That was just like our workaround, but that's really based on manual data curation, so scaling that approach up is going to be difficult. Now, what we did with that is that this theoretical biomarker data was linked to the data we had in the pathway models to make sure, do we actually have everything that's potentially changed? And then from the clinical data, we said, okay, which biomarkers are significantly changed, and can we link that information together and select pathways? And with metabolomics data, like I said before, statistics, not always a good idea. So I said, okay, I have 13 significantly changed metabolites. Out of all the pathways we have, how many of them are in a certain pathway? Then I had one pathway, okay, there are 8 in there. That's the first one I pick. The 13 that were significantly changed, minus those 8. I have 5 left. How many are in the next pathway? And I did that 3 times, created a top 3 of potentially relevant pathways. We visualized the data in Cytoscape and then gave it back to an expert that was blinded, so they didn't know which disease it was that they were looking at. Say, if you now see this visualization, can you actually make a diagnosis from that? And then this is one of the easier visualizations where the diagnosis was relatively simple. So you see uracil and thymine that are found in higher concentrations, and then a few steps below one is found, the blue one, in a lot lower concentration. Well, probably the enzyme that's not working is DPD or DPH or UP. It's one of these 3. And that was something that they were previously doing in their head. So they knew all the biochemical pathways and trying to map the data on the pathway, they just did that internally, not visualizing it. What we also noticed actually with this pathway selection is that we sometimes found some pathways and some metabolites that were significantly changed that are not reported as biomarkers. And because these diseases are so heterogeneous, those might be nice findings that you would normally not look at. We didn't have a lot of patients, right? These diseases are rare. And in the end, 3 cases were undiagnosable based on the metabolomics data alone. We didn't have the whole exome sequencing data yet at that point. Maybe in the future we can work with that. But this is in line with what they previously all did manually, except for now this whole pipeline is available and you put in the data. And if you have the data from the IEM base, that's the main caveat still, this analysis takes 30 minutes. Just to visualize it and interpret it, which is a lot faster than doing it in your head and thinking, which pathway is potentially changed here. But we also had some patients where the diagnosis was really complicated. And I also visualized that in the heat map. So this is with the theoretical biomarker data. This is the patient And this is how far the patient is away from all the theoretical biomarker data. It's basically its own little island, you know, nothing is linking to what is known for this patient. And what we actually notice here in this top 3 pathway selection that we did, so the disease is actually located in the urea cycle. But the problem with this disease is that there is a common metabolite between urea cycle and pyrimidine synthesis. So if you have a problem in the urea cycle, some of these metabolites actually get transported to pyrimidine, and then there are a lot of problems occur. So here this top 3 analysis didn't really help us because the best pathway match was just the 3rd option that we selected. So with metabolomics data alone, I do not foresee that in the near future this specific disease is diagnosable. There really needs to be something else to be able to diagnose it. And then this is the last part. I hope I'm still doing well on time.
You have 3 minutes.
I'm going to be super fast with this one. So this is the kinetic data model that we created. This is also available as a preprint, and I'm also working on that to get that published. And what we did here is also community creation. So we had students, we trained them in the Wikiparters Academy. They worked on these inherited metabolic disorder diseases, modeled it. And then we looked for kinetic data in 4 different databases that at that time did not have RDF or API. So again, typing something manually, putting it in an Excel sheet, and then visualizing that in Cytoscape. Now, this is the RDF model that we created from that, shows that we can link everything together. And I like to visualize things because that also makes it easier to write a Sparkle query in the end. And this is the pathway without kinetic data. This is the pathway with kinetic data. So what we see here is that only for 2 reactions we have data for humans. For 3 reactions we have data for rat, and for the gray reactions we have no data, kinetic data. Well, they also noticed, so we did a federated Sparkle query against a database with inhibitors. That's that pink circle. That's exactly the protein where no disease is linked to. And for all these other enzymes, there are no known drugs that can target this pathway. And that's the power, I think, of Sparkle, that we can actually do this integration with a lot of data and see where the gaps of knowledge are. And there are lots here, unfortunately. So if you're interested, again, we have another course coming up. This is organized by the BioSB community in Wageningen. So for the people that are here, that's maybe even better. In the beginning of January, and that is on knowledge graphs and data representation. And then you will learn to actually work with your own data and to represent that as a knowledge graph and to do federated queries and such. And that was it. Thank you.
So thank you, Denise. We have maybe time for 1 question.
Yeah. So thank you, Denise. I think at the start you mentioned something about sparse data, right?
Yeah.
And then you also continue to show how that we can overcome that partially by the linked data, by connecting things, well, the missing links through other modalities. You also mentioned community creation. So do you see ways how we can combine all of that to overcome that, I think, one of the most pressing challenges of this missing data by focusing our efforts somehow to...
Yeah, what I'm a big fan of is bottom-up approaches. So not a journal saying you need to do this with your data. I see that a lot in the data that's in the metabolite repository. People put it in and then the quality is meh, just because it's forced. So this needs to come bottom up. Everyone just needs to start doing this and need to start teaching people about it. If you have a PhD candidate in your group, tell them about all of this. And I'm assuming that the people that are here are doing that. But also other people. I think this needs to become part of the educational curriculum in general life sciences. It shouldn't be that we're the computational people and they're, the lab people and we're not talking to each other. That, I think, would be the next step forward. And not me recruiting students because they need credits, because they actually come to me voluntarily because they want to learn about this. Yeah.
Thank you.
Thank you.
Yep. Yeah, if you go. Oh.
Oh, sorry. Did I open a new tab?
Meanwhile, what's the microphone?
Yes, this one. I don't know. Which one is that? This one? Maybe.
Okay, hi everyone. I'm Egon. I struggled a bit with deciding what slides I wanted to show because of all the speakers before me and there being a significant overlap. So I composed some slides. I will try to rush through it. A lot of these things you saw in a lot more detail already, but there are some things that I want to point out. Let's see. So first, perhaps a bit of background, because this is a project that started in 2011. This was here in Wageningen in a building a few hundred meters that way, where we started using linked data and ontologies for metabolomics data. Unfortunately, there was a really bad year for me. This was part of the Netherlands Metabolomics Center, but my father died at that moment and I had to escape the Netherlands because the Netherlands is way too small research-wise. So I went to Sweden. But MedWare was continued. Stefan Neumann is listed up there. Who many of you probably will know. Yeah, so basically it took some time, but there is some really interesting things here. And one thing that we did, for example, we had a semantic model and we created user interfaces from that model, from the ontology. We translated the ontology into graphical user interface components, like forms like this. So that was actually quite fun, and that started a lot of things. In Sweden, I was actually more in the area of drug discovery there. Well, we continued using linked data approaches and semantic web approaches. One of the things that I did in Sweden, for example, is the semantic web version of Campbell, and I saw Campbell mentioned earlier as well. Okay. This is one of the more important slides here, I think, particularly with the discussion this afternoon here, but generally, and it was hinted upon already. This is a small dialogue from this book here. I started reading it. I don't know how it ends yet, but so far I like the book. And this dialogue is there are a couple of people in the library there. And it goes like this. This is where the next secrets will be found into the library. So Livera asked, okay, it was huge. Hundreds of rooms, millions of books. There must be an index, right? Well, Milan replies, actually there are many of them. Wait, what? Well, RPX explains. Each new librarian who gets controls knows that in order to keep that control, they need to have a new index. That is basically a linked data in one dialogue. We're coming up with new ontologies all the time. Ontologies being index, new databases, new identifiers, and we need to keep that together. And one main obstacle is there actually who controls it. So this is one reason why I'm very much a fan of open science, because that distributes the control and the power and allows us to really be creative. We saw that in Adriano's slide presentation also with his point about the 6-star for Wikidata. We have a community process. We do not have a small board of wise people that decide how it's going to be done. The community can play, they can study, they can experiment, they can continue. It means that we'll have many different indices, of course, but it will converge, just like all our scientific knowledge has been converging and continues to converge. And so that is the whole research process. Okay, so we keep this in mind because it comes back all the time. So where are we? Well, knowledge bases is something that I've been doing for a very long time. I just said that, and Wiki Pathways is one of them. This is the most recent paper that describes it. It has information of where we are with Wiki Pathways right now. Let's see, it's not the only knowledge base that we have, and I'll show some more as well. But these are two earlier papers of Wiki Pathways. This is Pathvisio. This is how we edit these things. This piece of software is very dated. But we don't have funding. We struggle getting funding for keeping it updated. But it is running, and there's a lot of interesting things there that address the answers of the questions that have been mentioned this morning already. How do we track provenance? How do we link things? Where is this knowledge coming from? Those kind of questions, and there are some hints at that. So over here we can see that we can link individual bits of knowledge in these diagrams to literature. And what I will show at the end actually is that the new version of how we represent this information in Wiki Pathways is a lot more powerful. And we're not actively using that, but I think that will change next year. It has to change next year. In the top right, you see in a new website with a new interface. We're in a transition phase where we have an old media wiki-based platform and something hosted with Markdown on GitHub pages, and with a lot more automation. Yeah. Let's see. Let's go on. So what about knowledge bases? Well, we have been using and linking Wiki Pathways to other things. We've been using it for rare diseases. We've been linking it to adverse outcome pathways, which is a new approach methodology used in regulatory sciences for things that are not safe for us, that are toxic, that have adverse outcomes. So this linked data approach is something that we have been using in a lot of different situations already, except for perhaps plant sciences, because Maastricht University, until recently, did not have a lot of plant sciences. We're using ontologies for that, and there are two ontologies that we maintain in our group, or well, three actually. The nanoparticle ontology, not on this slide, the chemical information ontology, ChemInf, and the inanimapper ontology, which is an application ontology for nanosafety. This is an overview of the knowledge bases that we actively provide as a service to the community at this moment. This is done by people in our group. Marvin is hosting a couple of them. Amar has been hosting a number of them. I have taken those over so far, and I am expecting that next year this list is going to grow, because the technologies to do these things, both creating RDF, both making more interoperable RDF, as well as the hosting, is becoming a lot easier. We're not entirely push button yet, but that will also probably change next year. So basically it will be a Sparkle endpoint for every researcher. We're very close to that. So I totally agree with other statements. Yeah, if you haven't played with Sparkle yet, if you haven't looked at it, this is the moment to start doing that, because it will only get bigger. Anything else? So this is the latest Wiki Pathways paper. You will read a bit about it, the new interface that we have there, a bit about the automation, but also things like the version control that we're moving to Git-based, archiving of pathways on Zenodo to make things fair. So it's really integrating a lot of technologies that we have for fair now. There are some things missing there, but I wrote that down in a document in preparation of this afternoon. I don't think there are any technological limitations left anymore. Basically, for any point where the question is, well, there needs to be some software that does this, it exists, at least in 2 or 3 different solutions that you can select from which you like most. It's not a technological question anymore. The use of semantic web technology, or the choice to not use them and just keep the mess that science in itself is really a choice. It's not really because it's not possible anymore, or that we need better tools. No, they exist. You may disagree with that. Let's see. So what are the other knowledge bases? Just to give an overview well, you have knowledge, you can represent it. The question about how do we represent uncertainty? We've done that. Actually, I gave you a presentation in my next group 8.5 years ago. That was 2017 already. And I actually went in that talk into how do we capture uncertainty in our experiments. And we have solutions for that as well. And this is actually one area where semantic web technologies do great because we can express it in the exact same language as we represent the knowledge itself. And this is the model, and we can talk about the model. We can express this model in the same language as well, so we can use that. We can reason over that. We can use all the chemical similarity, the evolutionary hierarchies of the taxons. We can all integrate that in one knowledge and use that. Another resource, this one and the previous one is really in the area of toxicology and safety. This is the Campbell database that I started in Sweden, as I mentioned, and I'm now going to pick that up again. I just need a bit of time figuring out how I load an SQLite database in Java and start spitting out triples from that. The code that I wrote for this was still written in PHP, but I don't think I can get away with that nowadays. I don't have to say a lot about that. Wikidata and Scolia, yeah, this is something that we have been working on in collaboration already. What I will say about that is that one of the reasons why I did that is the link of things to literature. It has a like mesh for biomedical literature. It has a very detailed keyword annotation, but at a semantic level. And that alone gives us a lot of opportunity of linking resources together that matter to the research that we're doing. I will not say much about this either. It was also mentioned. So the paper in eLife a couple of years ago with Andra Waagmeester. This one is interesting. That is actually more recent. You will see there Jasper Koelhorst, who is here in Wageningen as well, who's not here right now. But one nice thing is that we can enrich Wikidata when it's not complete. If it exists in nature, in the real world, it can go into Wikidata. And what we didn't have at that moment yet was identifiers for the proteins of the SARS-CoV-2 virus. So we actually minted them. UniProt didn't have them. They were months later. They have a longer process, but we needed them quickly because we had our pathways where we wanted to link literature to that pathway. So we needed identifiers for the SARS-CoV-2 proteins. We did that a number of months later. So this is 2021. So about a year later, we described how we did that and how we made sure that we had control over the data model and the way we pulled in that data. The article pulls in not just for the SARS-CoV-2 viruses, but also for a number of related coronaviruses. We've been pulling in other data. Protein complexes. That is a very a nice one, and possibly important for the biological pathways here. But I do not know. That is definitely biological knowledge that I miss. If the genes are closely related and co-expressed, do they actually form something like metabolomes? I do not know. Yeah, we have knowledge bases that we can link in this way. CAS registry numbers, those are traditionally still important for metabolomics. So I was happy that we could help them with updating their common chemistry resource. Lotus, very well covered. So happy that that went into Wikidata and that I could be part of that. An awesome resource. I really have to see how to get that Lotus bot working for other stuff as well. That posts things on Mastodon or social media so that I get to see or get some insight of what data is there. And I also like how it actually does encourage, maybe it's just Adriano and me so far, but edits in Wikidata after the post. If it's not on Wikidata, we can set up a copy of the software of Wikidata, Wikibase. We did that for one of our current running toxicology-related projects, and we can add additional information there if this is not notable enough for Wikidata, or where we just want to experiment a bit more before we start pushing it to Wikidata. So this is one of those resources. It works the same way. We have properties. We can add all the chemistry. I can add Wikidata identifiers based on an INCHI key match, and then I can start doing federated Sparkle queries, as Adriano explained. We can all visualize this in all sorts of ways, just like with Scolia. So I can run my Scolia on top of this Wikibase, just different Sparkle queries, but otherwise it works the same. It can link out, and this is one of the reasons for me to do a Wikibase here, because there are a lot of databases that I will want to link out, or resources that I will want to link out, that do not at this moment have identifiers, external identifiers in Wikidata. If I can show that there is sufficient use of those external identifiers, I can then go to Wikidata and propose the query. Because you need to show that something is notable there. So, what does this mean for plant modeling? We saw the biosynthetic pathways, and we have some plants in Wiki Pathways, not enough. I'm really looking forward to see what we need, which plant species. Adding new plant species is, thanks to this transition to GitHub Actions, has become a lot easier. It's still a manual work, but not in this diagram, but 3 weeks ago there was a Japanese biohackathon, and they had a project there on pathways again. I think the third year in a row. Alex Pico from Wiki Pathways had been attending the biohackathon in the past. And so there were a number of people there drawing caffeine synthesis pathways for species that we did not have. So I was remotely listening in on their biohackathon. So I added them to Wikidata. They're not released there because not all the genes and metabolites have identifiers yet, so I couldn't approve them yet. Which brings us to a second thing that, well, we still have them on the old website. We're still working out what to do with all these pathways that haven't been approved for the new website yet. But we can do that, and we have a number of options. We just didn't select it yet. So the list of species that we have on the old Wiki Pathways website is actually quite long. Well, relatively, it's not even close to the 4 million that we have in Wikidata, but that is a lack of knowledge problem that we have, I guess. This is basically an open question to the plant community. Which species do I need to add? And then you can start drawing plant pathways. You can add them annotate them with literature, and in this way, start collecting a bit of knowledge. That is all still with GPML 2013. Another thing that I have no idea. Oh, perfectly well in time. That I want to point out is the new GPML. The new GPML was scheduled to release in 2021. Well, the data model has been released in 2021, but the tools around it, partly because of the pandemic, they're not all updated yet. We have some tools, as we will see, but this is really interesting. So one thing that I think if we start doing this is we have to draw these pathways with this model. We have the tools for that. We do not have the whole platform to publish them on the website in an automated way yet, but we know how to do that. So something that will happen next year as well. So we will be only slightly ahead of the curve here. But the reason for that is actually that the new model actually has quite a few relevant things here. One thing that Wiki Pathways at this moment doesn't do is multi-species pathways. And with the plant microbiome interaction or the human microbiome interactions, these things are increasingly important. We cannot represent it in the current version of Wikipathways very well. With this GPML version, we can. We have to start using it, and then we can see what updates we need to make to our tools to make it easier to use, etc. We have better annotation of data sources, and probably need to use that one. So the cross references, the X references, is a lot more flexible. In the new system, we're much closer to things like identifiers.org. So that means that adding a new database, a database that we didn't have before, is a lot easier. It doesn't require releases of BridgeDB first anymore. A great improvement. We have a lot more information, well, that is a bit more the provenance of the pathway itself, about the data nodes. And one of the things there that we can have is everything is better referenced. So every data node, the data node can now be an ontology term as well. So data nodes so far always had to be something in a biological database or a biomedical database, like in gene or a metabolite. In this version, they can be ontologies, so that brings us much closer to linked data in general. Something that I think is very important. And the other thing that I was actually looking for, yeah, that's over here. That's change number 2. That is evidence. So far, Wikipathways has been a knowledge graph. But as discussed, we want to understand, we can't fully understand a knowledge graph if we don't know where that knowledge is coming from. If you see a contradiction, which we will increasingly see just by having more knowledge together, it's basically we're visualizing, we're making in a machine-readable way the scientific discussion visible. So we're going to have contradictions. And we should not shy away from that, but we should enable ourselves to see, well, why does this look to be a contradiction? Is actually one of the two wrong? Or is this just a different way of describing it? So is there some slight ontological mismatch? Or is there something else going on here? Is this something that we misunderstood in the past, etc.? So that evidence, that allows us to use the evidence and conclusions ontology, so to deeply annotate things like, hey, this metabolic reaction here, actually we never really measured that in human because it was measured in mouse or rat, and we just know that that protein with a high conservation is at present in human as well, so we're pretty sure we see both metabolites and we have the protein, so it probably happens in human as well. We can express that explicitly right now, but also things like the metabolite annotation. We can include that in these pathways. We do no longer have to shy away from saying, okay, we have a metabolite here. We're not entirely sure it's this metabolite class, or maybe it's this one because we have seen that in related species. We can annotate that with the proper evidence conclusion ontology. So later on, when we have that question, hey, that looks funny. How did they get to that conclusion? Why doesn't that match my data? We can actually see where that is coming from. So what is that pipeline? And I go back to that dialogue in the library some time ago. What we have right now, so we have our experiments, we try to encode that in literature, journal articles, the narratives. We're not really good at writing those narratives. They're human language, not very precise. We do our best, but they're not good enough for a number of reasons. It's a narrative. The journal articles are a story. It's the scientific argumentation that you describe there. As much as possible, you try to allow at least the reviewers to reach the same conclusion based on your new data, on your new evidence. But it's mostly the narrative that is practically being reviewed. Well, fortunately, we have databases that do a lot of curation after that. Not every journal article, even in the best journals, end up in databases because the database curator actually says, no, they made a mistake. Happens all the time. It does not end up in a database. journals that are indexed by databases like Ensemble, by Uniprot, that have a large curation team, that have articles that are not used as a reference in that article, are probably of interest, because they're probably not true. Because a curator looked at it and decided, no, this is not good enough for our database. We're not going to use that anywhere in our database. They're not publishing that data, but they have that data. But you should be able to reconstruct it. So that's that step. For the pathways, we have our literature, we extract the database, the pathways there, and with the new GPML we can do very far. And then we have our software to make it proper linked data after that. So that is that part is the current situation of the work that Andra Waagmeester did on making wiki pathways a proper citizen of the linked data network. Almost 15 years ago now, yeah, going in that direction. What I've shown here is a good bit of my own research, but I had a lot of collaborations. And this should be a pretty exhaustive list of all the people that I co-authored with. It's still, I'm not sure if you can still read it. It gets worse every time. But I particularly want to point out Denise, who you saw, Adriano for Lotus and Wikidata, Andra Waagmeester that I mentioned a couple of times because he did a lot of linked data work in the group. His work was part of the reason why I joined the research group in Maastricht. And Alex Pico and Martina Koetman, who are two of the main drivers of Wiki Pathways before. With that, I'm back at my opening slide. We have two questions. There are some bonus slides later on that are about retractions. So I leave that for a later moment. But our knowledge bases, they're not static. We have to monitor them. We have to make sure that knowledge that was derived from an article that was retracted in our database as well. And does that happen? Yes, that does happen. But we have our fair linked data approaches to solve that. But this slide and the slides that follow for that go into that part.
Thank you very much. So maybe one or two questions? Maybe one question.
Then I have a question to everyone here. Oh, and after that, I'll go first. So, about at one of the first BioSB meetings, at some point, it was a session. Andra and I were both there. It's probably something like 10 years ago. We asked at Bioinformatics, who here uses Sparkle? And at that moment, the conclusion basically was, and it was openly said, bioinformaticians will never use Sparkle. That was the message here. Can I see some hands? Who has actively used Sparkle? Okay, good. Who is in the side that they will never use Sparkle? Wonderful. I think that's a nice update, and we'll link that back there.
Yeah, so, well, thanks for introducing us into all these different things that you developed over the years, and nice that some of it was originated here in Wageningen. But now looking forward, right, especially for the planned pathways, where do you see, I mean, you mentioned some challenges, but maybe can you mention, like, for the next years on how can we really get to, like, a lot of planned pathways in wiki pathways?
Yeah, I think the bottleneck here is the first two steps here. Humans. Yeah. It's not a technical issue. It's just a human problem. Yeah.
So what should humans do?
Well, I would argue, and people are working on this. So the experiment directly to linked data, or at least directly to fair data, is and for example, I know the metabolomic community with MZML, those kind of things. They're very actively working on that. But making sure that the experiment, that the literature is not in between. So they should be parallel. This should be experiment to database and experiment to article.
Maybe then
that would already be a win if we would consistently do that. But we have to get rid of the journal articles for
Final
that.
question then, maybe do you see a role, because the first speaker, Louis Felix, he introduced Metabo, AI, and like LLMs, the use of LLMs. Do you see a connection there on how we can exploit that?
I'm going to disagree with you.
Please proceed.
No, I think I don't think LLMs are the way forward. Not at this moment, and I think the RECs and perhaps the MCPs, which I started, they're a bridge, but I think we're trying to solve something that we should not be solving. We would not need our English language to capture scientific data because it's not the right language. We invented mathematics, we invented chemical language because English language is not precise enough. And now with LLMs, we're trying to go back and say, no, we don't need scientific standards because we can all do it with English language. Now, you could argue that LLMs, and they're pretty good at it, they can learn some of those languages, and programming languages, they do pretty well, but those are quite small languages. But take, for example, IUPAC names, chemical names. Language-based AI models, they really struggle with learning these things. And I think that makes sense. Now, I don't think LLMs should not be used at all. I think they definitely have a role. I think they can be beneficial. For example, so you saw how much I was struggling with disagreeing about this. LLMs can help me with that. They can make a strong message easier to digest.
So thanks. I can conclude that humans are the problem, but for the time being, they are also definitely part of the solution.
Yeah, it's a choice. Do we want to push this forward, or do we want to continue with nice narratives but insufficient detail, causing all sorts of articles retracted at some point? No.
Is this an either or I mean, I like the idea of let's just ditch journal articles, but I guess you could also
No, I don't think it's neither or.
I guess you could also couple like RDF descriptions of data to journal articles, right?
At this moment, we have at least 100 of very useful literature that is not exposed in a machine-readable way, and this is definitely where LLMs will help us explore this literature. So in the foreseeable future, I think these LLMs will definitely help us. Moving forward, we have to get the experiment directly into databases. It will actually help all this AI to extract the extract knowledge from the past literature as well. Yeah.
Thank you very much, and thank you everyone for joining. I hope you enjoyed the talks, and looking forward to stay connected with many of you, or otherwise reach out.
Also, can we please give a really, really big hand to Elena, who organized this mini symposium.